Okay, we can start with people in Korea for the timeline. Richard, let me know if you have to go. We can do right after. Okay. Juhi, do you wanna give us update?
Yeah, not super much update. We just finished. Eugene submitted the two abstracts. So one on TOO and one on like the whole manuscript. Just like how we showed you last time, but updated. Uh, and now that we have more time, we're gonna go on to the actual manuscript. So just putting in PSA and putting in the comments. Um, but like I said, I worked on the remember reviewer 2 wanted us to like deal with a paragraph that said how like meta-analysis might not work because there's like all these like diverse essays that we're trying to like put into one. So I did write a paragraph on it. But it's like okay. I'll show you like next week when I have the PSA paragraph too. But like
Okay.
it doesn't sound strong, which is why I'm like I'm not really sure what to do. But I showed it to Liam, and I sent the link out for everyone to see. But I feel like that's the best we can do. But, I don't know. I might have showed it to you last time. I don't remember it. So yeah. We'll just finish up the meta part and the PSA. Just try to retouch everything, finish up the updates, and then show it to you next week.
Okay.
And after showing it to you, we're gonna send it to JCO because they wanted it in an email. See if they're still interested. If they're not, then we're gonna start the submission process with the other journals.
Okay, and we set those orders, right? I think.
Yeah, we did. We already did that.
Okay.
That's all for now.
Okay. Yeah. And I guess yeah. Also, these companies are, publishing, so we want to be most up to date as possible. That's why these papers have to be out, fast. Because we're
Yeah, I'm noticing that there's a lot of new stuff that are like they're
yeah,
showing their interim results. So before they show their final results, we
yeah,
need, yeah,
yeah. I think interim we're fine. It's not final, so but
yeah. Like
yeah.
interim, like some of the results looked iffy. I'm guessing because it's not complete,
Mhm.
but if they send out the complete results, that's gonna be yeah.
Yeah.
So I get what you mean. It seems everyone's very interested in this.
Okay. Good. And I went through the ASCO abstract last time, right? Was it?
You did, you did,
Yeah.
you did a word by word.
Okay. Did you submit?
Yeah, it's submitted. Both of them are submitted. So
Okay.
now we have time for the actual manuscript.
Okay.
Yeah.
So yeah, let's get it done. When was the birth of this project? When you
I
came?
don't,
That
I
was
don't even know. Like a year almost.
almost year.
Yeah.
Yeah.
I think I started like April-ish.
Okay. April-ish.
Yeah. And it's like February.
So it's like really like it's a big project. It's an ambitious project.
We
But
kept adding more stuff.
and then it started as review and then it got to meta-analysis
I know.
and then yeah, and then so many
We
different
did the
like,
subgroups, the
yeah,
TOs.
yeah, and the tissue of origin became
Yeah.
a good time. So this is a good lesson where like this is how the project evolves or
Mm.
turns into the next question and but the question itself is very important. Like, how
Mm.
can we best screen general population for early cancer detection? You know, is it worth it? Is it, you know, scientifically valid?
Mhm.
And who determines it, right? Like what's the, you know, what's the reaction from medical society? And so, you know, it's a very important topic, and we haven't seen like the most comprehensive approach yet. So
Mhm.
we're trying to take the first step. So that's
Yeah.
okay,
So I
good.
definitely see that they're interested, and I definitely see that it will get published somewhere,
Mhm.
even if it's like a less prestigious journal because of the method that we employed, because like a lot of them don't agree with it.
Yeah,
Um, I see it's gonna get published. I'm just trying to make sure it's updated.
yeah,
And make sure we go down that list instead of just going to like an easy journal straight up.
right.
So
Make sure, yeah, that's really
yeah.
important. You know, unless you try, you never know. And sometimes you get, you know, a surprise acceptance. Sometimes
Mhm.
you get it without like reviewers like praising about it and then no real big comments. So
Mhm.
it's really unpredictable. But I'll tell you for all of you, there's a lot of conflict of interest. You know, a company funded by angel investors, and their job is to protect their own essay, their own company, right? But
Mm,
if you're comparing it to other and then just combining it with, you know, it's not good for the company. So those
mhm. Yeah.
papers that have come out, the authors are going to get the review request because who is famous in the field, right?
Oh,
That's how reviewers pick, the
yeah.
journal picks reviewers who
Mhm.
actually published first in that field. So we'll go to that company or a professor who worked with that company, right?
Yes,
So they're going to have their own agenda. So they're going to try their best to block it.
that makes sense. Yeah.
So yeah, that's what happens. If there's a lot of people with different interests, and
Yeah.
that's normal. That's, you know, that's what it is.
Mhm.
You just have to find a right reviewer that will appreciate the value of this kind of approach. So
Mhm.
yeah,
So,
it's not a matter of right or wrong. We just have to be, you know, thanks to help of Dr. Shin, just, you know, methodologically rigorous, but
mhm.
philosophically, people can be on different end.
That
Uh,
makes
so
yeah.
yeah, I just want you to understand that.
Yeah. That actually makes much more sense now that you say you told us that, because I guess I didn't connect those thoughts in my brain.
Mm,
Yeah, that
yeah,
makes a lot of sense. Yeah.
yeah, yeah, yeah. Okay, but good job. Keep doing it, and let's just
Yeah,
continue
everyone's
to
like really
submit it.
working on
Yeah,
it. Yeah.
yeah, and yeah. Let's see what ASCO says about these.
Mm-hmm.
ASCO is very difficult to get accepted. It's very prestigious. So
Oh, okay.
yeah. So let's see.
Okay.
All
Sounds
right.
good.
Thank you. We'll go to Eugene.
Yeah. Um. Hello. Thank you for giving me the you know chance to present first. I have a few things to be confirmed by you. Um. First one is hyperprogression. I know it started long ago, but I think I finally finalized the tables and the figures. So it would be great if you can just have a last look at them and confirm. Um. Can you see my screen? I think you can, right?
Yeah.
Yeah, so I think I just made it too many tables and figures, so I just list all the names and yeah, basically names, the legends of tables and figures like this. And table 1 is clinical and pathological characteristics of patients, and table 2 is about incidence. across different definitions and modified definitions. And 3 is prognostic factors associated with PFS and OS, univariate and multivariate analysis. And I think I can combine tables 4 and 5 because both are dealing with the risk factors, whether it is clinical pathological factors or genomic alterations. I'll update you with this table 5. I just did the analysis. Thanks to Dr. Jian, she provided the genomic alteration data last week. So I think I can combine these two. And then supplementary table 1 is, it's about the ones that are discovered as having hyperprogression by modified definition. So we are confirming that they are not having like pseudo hyperprogression. So that's supplementary table 1. Supplementary table 2 is univariate analysis result for PFS and OS, linked with table 3, I think. And figure 1 is Venn diagram, and figure 2, I try to make a new figure. I'll show you later on. I think it can be going to supplementary figure 1 as well. And figure 3 is kind of, about new lesions like Wang did in his paper. And figure 4 will be kind of main figure OS and PFS according to PD, HPD, and PD without HPD. So I'll just briefly update you with totally new, which is table 5 and figure 2, I think. And so this is figure, this is figure 5. It should be 5. And so Dr. Ji, according to the data collected by Dr. Jian, like these are genomic alterations that each like different groups are bound to have.
Uh-huh.
And I try to make the table that is pretty similar with one was in like Kato's paper that you introduced to us. So although like frequencies are pretty low, by logistic regression, like the numbers are, because of the low frequency, I think numbers look not reasonable, but weirdly, like p-value was significant for some alterations. For HPDC, Champia, SMARCS CA4 was associated with hyperprogression, and for Sarabusit, BRC2 and RED50 was found to be associated with hyperprogression. By definition, I think we cannot argue like strongly with this seemingly exaggerated numbers, but at least these are results. And the reason why I want to combine this and the table, the other risk factors, I think we can at least do like multivariate logistic regression to see whether these kind of significant correlation will be remaining or not when we combine kind of genomic alterations data and then other like tumor burden or what else, like tumor stage or other histology factors. So what do you think about this? And I realized, so first of all, when I try to analyze risk factors, including these clinical pathological variables, I try to do like Fisher or chi-square test, but I recently realized that I can do logistic regression even for like risk factors, like I did for genomic alterations. So I'm trying to do logistic analysis for these variables as well. So I'm trying to combine table 5 and this should be 4, so 4 and 5.
So here's what I think. Here's what I think.
Okay.
The number of mutations found are so small, and it's only, I guess, so for out of all the patients, how many actually had NGS?
I think more than 80%.
So it's important to put. So 4 patient and 117, they all had NGS.
Oh, I made a mistake. So I don't think so. I think the denominator should be for these columns. Denominator is 4, and I think yeah, I'll check that out. I think at least 8 or 10 patients, they didn't have any like reports on genomic alteration, so I kind of made them into not available.
Hmm.
So this should be somewhere like 110 or yeah. I'm sorry. I think this percentage should be
Yeah.
changing according to yeah.
So
So are you okay just with kind of list the alterations rather than reporting the frequency? I know frequencies are too small.
yeah, I mean, if you look at SMART, there was only 3 mutations. BRCA2, there was only 2 mutations
Um,
found, right, out of 110 patients. So what I'm
right,
saying
right.
is the frequency is so small
Mhm.
that it doesn't make sense to present it this way. So I would report that in the supplemental table.
Mhm.
Because the numbers are so small, I don't
Mhm.
think doing odds ratio for this makes sense. Just do a chi-square p-value. I think it's good enough.
Okay.
Yeah. And for table 4, I think it makes sense. It's okay to do odds ratio. And but
So that means it's okay to make table 4 into a format of table 5, right?
yeah,
Or like this. Okay.
but you have univariate and multivariate there, so you don't have to do multivariate
Or like risk factor, right?
because it's a lot.
Yeah, but I the reason that I make the table like this is I just copied the format of the ones that are in the Kato's paper. They did exactly the same thing for genomic alterations they reported, and I mean I noticed that even their frequencies are pretty small, like not this small, but like a little bit more.
So
And yeah,
Kato did report. What was their total number?
total they got information from 150 patients, and but then frequencies are like less than 10% for each variance. I think I cannot remember the exact, but so even for them, they were not able to find any kind of significant um genomic alterations that are associated with risk of hyperprogression, but they at least did report frequencies and all the, and then they did some kind of some other type of analysis given that it's, frequencies are too small, I think, but I honestly don't know how to do that. Like, it's called bootstrap analysis, but yeah, if you want me to like learn how to do that and do that with our data, I mean, I can try.
No, it's okay. Yeah, I think the numbers are small. It's hard to make. But at
Like,
least, SMART 4 out of 3, one was in hypertension. Um, you know, so 25% versus 1.8%. You could kind of describe that in your result text.
okay.
But for table, you can make it supplemental.
Mm-hmm.
But
Yeah,
where is
understand.
MDM2? Not 0?
Sorry, could you
MDM,
repeat? Oh, MDM2. There's no MDM2 among like the whole patients.
yeah. Okay. So
Yeah.
we could, you could say all our hyperprogression, we did not find MDM2 in your discussion because that's the one of the important gene that we're talking about. Yeah.
Right. Okay. I have to mention MDM2.
So here the resist go up to the
Okay.
tumor burden.
Yeah,
Baseline tumor burden was smaller.
smaller in non-hypertension patients
Hmm.
for Champia definition, not for Sarabut. I think it's tricky to discuss because trend were different between two definitions. Um,
And pre-baseline.
it's also yeah, smaller in
Hmm. So in your literature search, did you
uh
find any evidence bigger
uh
tumor tend to have more hyperprogression?
not until now. I'm trying to, but uh, at first I try to search any literature supporting like tumor size at baseline or pre-baseline is related to more aggressive characteristics, but I was not able to do that, and I didn't find any literature supporting that. And for hyperprogression, like, yeah, I was not able to find any literature
Hmm.
up until now, but I'll keep trying to search.
Hmm. Okay.
Okay.
But just,
Okay.
yeah, whatever you find, describe that in your discussion, that.
Yeah. Yeah, you mentioned that a few times. I'll keep that in mind. Yeah. And okay. Then and for the figure, I made I revised Venn diagram like this. This will be the final version. And the figure that I remember that you kind of try to draw something like this. And the last time you gave a comment that it would be great if we can have real time difference, time gap for each patient rather than just the relative time gap. So this is so among 121 patients, 4 patients were found to have Champia hyperprogression. They're here. And if we modify our definition, if we include new lesions, new targetable lesions, then the incidence went up like this. And even for the previous ones, because we included new lesions, the slope changed. And actually the axis is pretty similar, so you can compare the slope. For this green, there's no new lesions, so it's pretty similar. It's exactly same, but for the red line, this patient has new lesions, so it went up. And
So
so Yeah,
So the left one is the regular resist.
yes. The kind of original definition, and right side is modified definition.
Okay.
Um,
With modified definition, you have more patients who meets the
definitely, yeah,
criteria.
the criteria. But the problem of these figures is that it kind of indirectly shows these patients are having hyperprogression rather than directly showing them, um, you know, directly like giving us some parameters that we can notice that these are hyperprogression. So I kind of try to compare TGR ratio. So if you look at this, for original one, TGR ratio equal to is dotted like this, the line, and the blue one is the blue one, like the patients marked with one, they are the ones that are found to have hyperprogression by original definition, and the other ones, color coded as red, they are found to have modified hyperprogression. If you look at here, the modification line, but originally they are not meeting the criteria of TGR ratio over or equal to. So I think it's okay to present this rather than the the ones that I show you earlier.
So original versus modification, I don't know if, uh, um, so all of the lines meet TGR hyperprogression definition. Because the ratio is 2 or above, right?
When we modify them before modification, if you look at close, like red dots are below the line, below the criteria, so they are not meeting the criteria of hyperprogression. But when we
Oh,
modify,
I see. TGR ratio 2, I mean, that's okay, but I'd rather on the y-axis put number 2 there, so.
yeah, that's what I try to do, but I was not able to do that, so I kind of did this alternatively, but we'll figure
Yeah,
out
yeah,
how to do it.
put number 2 on the y-axis as well. But anyway, I think this is nice graphic presentation. TGK, same thing.
Same thing, yeah, this is from Sarabu's definition, and yeah,
So
so
TGK, I think some slopes going down.
no, it looks like that, but I checked the data and then they kind of they are same
Same?
because
Oh,
yeah, because they have no new lesion. So many of them doesn't have new lesions among the patients who were defined to have Sarabuji type progression by original definition. So yeah,
your question is, should we show this or versus the other?
yeah,
Right.
like this is for Champia, and this is for Sarabuji. It's more patients and more colorful, but I mean I can
Oh,
change. Yeah.
so for the color, so you can put, you know, you don't have to put, patient. I think different color, you can put the, oh, I guess different colors are different patients. I think they understand. Uh, so yeah, these are very detailed, so keep these for supplemental figure. And if you, I don't know if you have room, do you have room for more figure?
I think for supplementary figure there's no limitation.
No, I'm
As I
saying
understand.
for the ratio, 2 graphs.
Oh,
For this one,
like like a main figure.
Yeah,
I actually don't know the limitation of the figure
so yeah, so
as
look
of
it
now.
up. If you can put it, if you cannot, then put all these in supplemental, but if you need, if you can just put these two for main figure, that would be good.
Okay. So like the name would be something like this, right? Or do you want me to combine these with the Venn diagram figure? Or keep it
Yeah, you can combine it and make two panels, figure one
right,
A, figure one B.
right. Maybe, yeah, okay. Because it's comparing like these patients, so okay, I'll do that. Um, but you want me to keep this detailed figure for supplemental.
Yeah, and
Okay,
just don't call it modified because it's so general term, modified. In the method, there's gotta be a way to, it has to be a specific term.
mm,
So original resist, right? You
yeah,
can put
okay.
modified,
Or kind of resist including new lead. Mm,
a modified resist.
yeah, I understand. This is not specifying what we did exactly. We included new lesions.
Yeah, that's the only thing you modified, all right? So,
Right.
yeah, maybe just call it modified resist
Yeah, and
and
then
make
also
it consistent all throughout your manuscript.
right. Modified
You
resist.
say
Okay,
modified, it's not clear.
I know it. Yeah, true.
And then in the method, you said when I say modify resist, this is what I mean. So in the
Mm.
method, you describe it.
Okay. Yeah. And actually, like the TGR ratio, I kind of removed the very like the outlier. So I'll just describe that into like in the legend maybe or so there is a one patient who have like too many new lesions. So TGR ratio went up over 200 from like below 2. So I think this graph is too, I don't know, I think it's not reasonable to keep graph like this because it's so hard to distinguish other dots. So I kind of remove this patient outlier, and then that's the way how this graph um, came. I'll just mention that in the below. And you think it's okay to do that, or should I keep this? Um,
So I think when you're trying, well, one is TGR, one is TGK, right?
yeah, but I'm trying. So TGR, there are two graphs that I made. So this is the original one. If you look at the y-axis, like definitely we can call this patient as having like being outlier. When we remove this patient, the axis kind of is shortened like this. So it's more kind of easy to distinguish like the remaining dots. So
But I think you can't remove one patient because that patient is also included in this study. So maybe you can use two wave lines.
So a lot of
um
times we do this. If you can see, you cross, you You,
Yeah, yeah.
you put some
Oh,
breakage.
I'll edit. Yeah, I'll edit the image. Okay.
So 60 and they'll go straight to 200 by putting a line or something.
Okay. I actually tried to do that with R, but I wasn't able to figure out the code to do that. I think I might try to just do it manually. Like,
Yeah, just do it manually, cut and use PowerPoint.
right. Okay. Yeah, I'll do that. Okay. Then, and these are the kind of alternative figure that I try to make, but I think these are good, right? So I'll just remove these and yeah, other than that, figure 3 is about new lesions. So I showed like number of new lesions and this is the ones that I showed you last time. So we kind of compare the modified ones into like having more 3 new lesions, more than 2. And then the last figure will be like this, just showing the association between hyperprogression and then OS and PFS with hazard ratio. Yeah, I'll try to make um, this figure 4 as like this. A Yeah, I have to make C and D, but
So,
as of now, like yeah,
so show me what it means.
what it
Range
means.
is
Okay.
no progression at
Oh. No, yeah, no progression at all. So
red is
green
high
means
Champion.
yes, and blue means like progression by RECIST, but not meeting the criteria of Champion. Oh, I'm sorry. I think I kind of put hazard ratio wrongly. So, but one should go up, and so based on the results, Champion has 5.6, you know, increased risk for death compared to patients who don't have any progression, and patients who have resist progression but not Champia hyperprogression, they also they yeah, their risk is also increasing. But between them, there's no
Okay,
okay. So
so this is OS.
this is OS for Champia, this is OS for Sarabusit, and I'm gonna make
Yeah, this one's wrong
PFS.
too. Yeah.
Yeah, yeah.
Okay.
Okay. Yeah, this is the update for hyperprogression. So as I'll just modify a few things based on the comments, and then after that, yeah, I think I could write up all the results and methods pretty quickly. Um, it was kind of hard to, just fix which should be where. So yeah, and other than that, like I have one other manuscript that is pretty done by Dr. Hyun Choi. Um, but I think there are a few things that needs to be done. Definitely, like legends, figure legends, or manuscript itself is yeah. There should be something done. So you do you want me to show you this one when it is prepared for like submission, or are you okay with just looking over this as of now?
So did you submit this already or not yet?
This one or this one?
This one.
This one, Dr. Choi just sent me this, sent me this today so that I can go over it. I don't think he has submitted it. He says it's pretty done, but Yeah, but I noticed a few things that needs to be done more.
Oh, this is the manuscript?
Yes, this is the manuscript, but I'm gonna put like citation and try to find some more citations as well.
So the title, I don't like it because it doesn't talk about till.
Okay, I think we should be more specific. It should be more specified, like
And then abstract. Uh, can you send the link so that I
Yeah.
can
Yeah, sure.
scroll it? Can send to chat
Yeah,
box.
sure.
Okay. And unshare.
Okay.
Okay. Do you see it?
Yeah.
Okay. So here, the deep learning based IP may play a role as a novel categorization of play, provide insight into discovery of a predictor. Provide insight into discovery of. I would just say may play a role as a novel categorization of. I mean, it just is mouthful and it's just so a lot of words. I always like to write it simple.
Yeah.
It like may help categorize tumors. Uh, and uh, and so why are you categorizing tumors? I mean, that's and provide, it doesn't explain anything, but so, I mean, categorize it, we categorize for better prediction. So it's the same thing. You're saying the same thing again. So
Right.
deep learning, IPs, phenotypes, may help better predict outcomes of endometrial cancer patients receiving immunotherapy.
Okay.
Mm. This is Lunit wrote this, or who wrote this?
So Dr. Horan Choi said he tried to write this part, but he think Lunit should have kind of overlook, and then they have to kind of write it up a few more. So we'll reach out to them.
Hmm. So I would say when you say this is results, so you can't just say the results are available. I would say the frequency, how it was distributed, what's the mean and range. The descriptive statistics has to be here. When you say similar tendency, you have to say, show what's the hazard ratio and p-value here as well. I know it might be in figure, but it has described. Use a special character.
Right.
Immunogenic is a very crude term. How do you define immunogenic? So you can say, well, inflamed is immunogenic. This is not the right term. So what did you actually see? Did you see infiltration of more
like macrophage or
immune cells?
yeah, I think here, yeah, there are a few more immune cells are there in immune IP.
So we further analyzed the gene expression. And where's the result? There's no result. Like what gene was expressed at what level, what's the p-value. And here's
I
immune
think-
check inhibitor checkpoints. When you say upregulated, this is not enough. This is really vague. Like what was upregulated at what level, and what was the p-value between different types. This is not the quality for result section.
Understood.
And if you need something in the supplemental figure and the graphs, things like that.
Okay.
So this is the common mistake you guys make. This is still in the results section. So results section, we're not talking about what other literatures have shown. So there shouldn't be much citation. If you want to describe it in the method, you can put a lot of citation in the method. This is what we did, what others have, we used the same method used in this paper. Here, we added POL-E genes based on this paper. Then this is method. This is not result. So in the method, you say when we are looking at mutational, landscape. But I don't know. Everyone knows POL-E gene is important in mutation, mismatch repair, all that. But my point is these describing method or literature has to go to different section, not in result.
Understood. Yeah.
So endometrial cancer patients are defined by different mutations, and there's usually 4 different types. And the POL-E mutated type is 1. then you describe that in your method, as proposed by this paper. We also classified endometrial cancer into 4 different types, and POLE gene mutation was important subtype. So that's how we did the analysis. And you put citations for it in the methods. And in the result, you describe, so what did I find in our analysis?
Okay, understood.
So this, to me, reads like an introduction or discussion.
Mm,
All you have to say is what you found. Inflamed IP had, higher frequency. When you say higher frequency, what is the percentage? What's the p-value? So you have to be really, really descriptive.
descriptive.
So again, POLE mutation is a better prognosis or not? This is all for discussion. So you describe anything you did in your results, nothing
Yeah.
else.
Um,
The same thing, if you have a heat map, show the graph, like enrich, like we need to see, visualize this pathway. And if there is any number, like enriched, what is the number of enrichment? Because usually if you do GSEA, there's a score or p-value. How tightly is this pathway related to that phenotype? It's
okay.
very vague. Three new subtype of tumors have distinct immunological mutational characteristics that reveal significantly different survival outcome. Inflamed IP prong compared to with non-inflamed, independent of MSI status. Inflamed IP still showed better in. I would say still regardless or irrespective of than in either, like which may be a clue to predict responder. So this is a leap of faith. We don't know. So in discussion, you summarize your best finding
Mhm.
and say what was so the question, the always ask this question, has this type of analysis ever been performed in the field of medicine? If the answer is yes, you have to say this is the first to report. The TIL based, so and emphasize TIL.
Okay.
Like just don't say we classify. Anyone can classify based on something they claim to have novelty.
Mhm.
But this is the first AI-based, TIL-based classification that ever showed um, at least prognostic implications in endometrial cancer. And we are the first to show that this has prognostic implication regardless of the MSI high status. So I don't know if it's first or not, because I haven't done the literature search.
Mhm.
So if it is really the first,
Yeah.
say we are the first. Okay,
Okay. Makes sense.
and make sure Horion watches this editing, okay?
Mhm.
A lot of sentences are, I think it's long, it can be shorter.
Mhm.
Our results seem to support this hypothesis, given that TMB, which may be surrogate for increased tumor, were enriched. So you know how this sentence is bad, like there's so many things. So I want you to try to write sentences very short. A leads to B, therefore, B leads to C, Eh,
Mhm.
if you read it, the idea has to come to you very clearly.
Mm. Understood. Yeah.
So the first step is get rid of unnecessary wording. Collectively, our results seem to support this hypothesis, given that, which may,
Right. It,
so you can
mm,
say, our results support this, and then you can,
Right.
and then so just maybe surrogate,
Potentially. Therefore, like.
so the notion is, let's say high TMB, high MSI high had more, I mean, people knew these groups have inflamed more T cells. It's very, I mean, and a lot of studies have shown that. Our studies using the AI based, again, TIL based phenotyping, also supported the same association.
Mm.
It doesn't have
Right.
to be seen to support the hypothesis. It's not even hypothesis. Most people know that MSI high, TMB highs have more TILs. So literature have shown that, and our study shows that too, period. So we don't have to, mumble fumble to make, to say that. You just have to make it what do you want to say? So just,
Right.
so sometimes it's not about grammar or the facts. It's about how you present, how you write it so plainly and succinctly.
Mm.
Uh,
Understood.
so you talk about different pathways here and there. And you say, finally, we compared immune-related gene expression. So again, I want more clarity. There's increased MHC, increased STAT3, increased IL-6. Then you have to say what happened to our data set. Like, what was their level? What is our level? Is it same or similar? What did our data show about STAT3, JAK2? It's probably JAK1 or 2. You have to say when you say JAK, it's 1 or 2. So what was theirs? What did they show, and what did ours show? Was it similar, different in level? So
compare.
this reads more like what others found, and this is what we found. CD274, which is you should at least put in the parenthesis PDL1. Some people don't know what 274 is. But anyway, it has to be more like, this is what the literature said, this is what our study showed. And is it same, different? If it's different, why is it different? So that's the whole idea of literature search. I don't see that happening much. So it's just clinical significance. So this can be redundant. You can just say, EC with POL mutations was associated with improved PFS.
Right.
And just cite. You don't need the sentence.
Right.
So about each paragraph, see, so summary and then literature comparison, talking about different pathways. Again, I would write a short sentence that summarizes the main idea of the whole paragraph each time. See how the sentence starts with keynote trial. This is a supporting fact. Do not start your sentence with supporting facts. So what's the main idea? It has, it goes for all other paragraphs.
Understood.
Why did you underline this?
Actually, this is done by Dr. Choi. Yeah, honestly, not knowing the reason as of now, but
So such as patients with low TMB absence. of MSI showing good response or vice versa? What is vice versa? High TMB showing bad
I guess
response?
so.
Is that vice versa?
I guess so.
So again, this is also a bad example of so what do you want to say? Wide range of response rates, such as patients with low TMB showing good response. So that's not a wide range. When you're saying good response, it's very vague. So here's the thing. We know the trend that high TMB, high MSI tend to correlate with better response. That's it. That's all we know. However, what we're trying to say is even if patient has no MSI high feature, low TMB, we do see responders. It's not a wide range of response rates. There are a few responders, and we don't want to miss them. And how can we better identify them? Is the next question. Is where AI-based, TIL-based IP classification may help in answering this question. So that's kind of the logic we're trying to say. But when you say wide range of response rates and it's hard to grasp what the writer is trying to say. It's not the unpredictability. It's a limitation of each biomarker. It's a limitation of TMB and a limitation of MSI high. None of the biomarkers are perfect. Every biomarker has limitation.
Right.
We were not able to assess. This sounds so generic. Understand tumor characteristics established. I mean, of course we know that, but this is not a counter argument for all the limitations. I think you can leave limitation as it is. We don't need this cliche here. So maybe look into. I know some TCGA cohorts, they do have PD-L1 immunohistochemistry data available, but it might be an issue of if you have 500 patients, that IHC data may be available for less than 100 patients or something like that. I don't know. We have 0% of IHC data for PD-L1. So look into it before we say it, because everyone knows TCGA data very well.
Okay.
So limitation. First, this study is a retrospective study. Second, so retrospective study is not a limitation. People do a lot of retrospective analysis.
Right.
So you cannot say this is a limitation.
Makes sense.
There are a lot of retrospective data prospectively collected. And some cohorts were prospectively collected. So this goes to any TCGA paper you guys are writing, maybe Dr. Myung-woo Nam, same thing. If you're writing. This is endometrial. So Dr. Eugene, I don't know what happened to the paper Dr. Hwon Kyung was writing. Do you know
I
what
know
happened?
she was honestly not knowing as of now. I can reach out to her. Like
Yeah, because
she was doing
writing a
ovarian.
paper in ovarian cancer and
Right.
endometrial cancer.
Oh, is she also covering endometrial as well? Not just for ovarian.
She did both.
Okay.
I'm just thinking, there's so many papers are lost if they start their residency and they stop writing it, and you know, it's just so hard to keep track. But let's see what, where she left and anything she presented before. Um, but anyway, she looked at the TP53. I think same thing was Dr. Jinyoung Hwang. She did endometrial cancer and looked at the importance of P53 mutation. Dr. Hwon Kyung and Hwang Jinyoung, they all did poster presentation and all that, and never published any of those.
Yeah.
So let's
I wish
follow
I
up with them and see where things are and whether they have
Sure.
any thought of publishing it. Otherwise, we can review what they have and just have them send all of their materials to you, and we can reassess whether this needs to be published or not. We can reassess that.
Yeah, sure. I will reach out to Dr. Ha and Huang
Dr.
as
Huang,
well.
yeah,
Yeah.
yeah. But it's good that Dr. Horyun Cho is trying to publish it, so.
Right. He's very persistent. Yeah.
Yeah. So for those of you, this is the first time that endometrial cancer was analyzed with TIL. And Dr. Horin Choi went to AACR and did oral presentation on this finding. And it's very prestigious to do oral presentation as a second-year resident. Okay,
Easier, right?
yeah. Anyway, but you have to write it. You can't stop it there. So for me, the obvious limitation is that these data were the limitation is most of them are early stage rather than late stage. So it may be difficult to extrapolate the findings to late stage. Number two, if it is true that PD-L1-IHC is not present in then that's a limitation. We want to know the protein expression of PD-L1, but and then you can say we were able to use the gene expression level of immune checkpoints.
Right.
And then number three is we don't know. Oh, this only shows prognostic implication. We don't know therapeutic implication. We don't know how they will respond to immunotherapy. None of them are treated with immunotherapy. So we cannot make that prediction here. This is not predictive. This is only prognostic. So those are, in my mind, main limitations.
Sure.
Yeah.
Thank you for giving us input. Yeah.
So again, in conclusion, I would say, emphasize that we're the first group who have done this for the first time. may be beneficial to identify responders. Suggest the new classification. So the way it is written, I don't like it.
Right.
So
Yeah,
our
can be improved.
our suggested classification, may help better predict uh endometrial cancer patients treated with immunotherapy or something. Just write it more plainly.
Okay.
It's predictive value to ICI. I like the expression. It's evaluate its predictive role in identifying responders. In endometrial cancer. This is mouthful as well. You can say this, our new classification can be used to complement other biomarkers and may lead to a more more robust predictive algorithm
Yeah.
in endometrial cancer. Something like that.
Great.
So definitely invite, I don't know, maybe Haesung can help refine some sentence. Haesung or Liam, whoever, or even Juhi, whoever can help in terms of making English expressions improve.
Sure.
And I want to emphasize, so for editing, if it's typo, grammar, they do not qualify to be a co-author. The co-author has to significantly impact the contents of the research. Okay, they need to go into the acknowledgement. This is an ethical issue. Okay. If they help in terms of logic and rewrite a lot of sentences and help with the contents a lot, then the first author decides whether the patient whether that person can be qualified as a co-author of the paper. Okay.
Understood. Dr.
But-
Choi is definitely first author, so yeah.
So definitely work with another person to
Right.
better write the manuscript. Okay. Okay.
Mhm.
Figure 1.
Yeah.
So ABC, like how
How is different?
how
Um.
I don't know how.
Oh yeah, it's not labeled yet. I need to label it.
HIP, MSI high. So I know you wrote it here, but it's also better to label it here so that people, when they see, oh, this is all comers, this is MSI high, this is
Right.
MSS.
Low, like MSI low.
PFS, immunophenotype,
Yeah.
mutational landscape. It's good that neoantigen is here as well. So yes, they are different.
The table, I think we're still deciding whether we need to have the other table or not. So yeah, it can be removed at the end of the day, but
Okay. I think you need table.
like a basic demographic or characteristics.
Yeah.
Right. Okay. Then as a main
Yeah,
table.
so you
right?
want to know, are these patients older, more male? You need, some, because you have patients, these are patients. So you need every, demographics, but also, pathologic characteristics. You definitely need the basic table here.
Mm, makes sense.
So ones that are significantly different, you have to write it out in the results. What are the, immune genes, and
Mm,
not just a look at figure. Because it's so small, we don't know what's important. You have to, if you think those are important, write it out in the results.
right. Correct.
Same thing, just don't say, oh, they are different. Like if there are pathways you think they're important, say it out in the result. These pathways were highly upregulated, highly expressed. Um, okay. So nice start, just still a long way to go. So next time I want to see a publication quality. Okay,
Okay.
no grammar
Right.
issues. Okay,
Understood. Yeah.
okay, but nice start, and we need to get it out. Um. um, So best next step is let's say if I'm an ovarian cancer expert, I need the randomized clinical data. I need the trial data, everyone treated with immunotherapy, and see if this signature separates the curve. And then if it separates the curve, what's the next step? I'll design a trial, enroll only the patients with the inflamed signature, and I'll give them immunotherapy. And the response rate is going to go from 10% to 50%. Do you understand? That's how we change clinical paradigm. So we have to get there. And as you can see, not many researchers are spending time analyzing data, and so you'll be surprised that research field is always wide open for those who ask questions. It a lot of times it's the bandwidth. We are always busy with our own life issues, and our next step, our next clinical duties. So it's hard to do research, but asking the right question and getting connected to the right technology it's a blessing. It's just appreciate what you have, and when you've done the analysis, write it out. Otherwise the world won't know. No. So nice start. So keep working with Dr. Choi. Anything else,
Sure.
Eugene?
Yeah, I'm done for today.
Okay.
Yeah.
And
later.
don't forget all your cancer incidents and
Right. I'm working on it.
depression, transfusion. I feel like you have a lot on your plate, but also um you have time to yourself. You have time to invest in research,
Right.
so make every use out of it. Because if you start clinical rotation, it's going to be very difficult to finish.
Agree. Yeah.
Yeah, so Yeah, you still have many months, but it really goes quickly once
So
you
yeah,
decide to
know.
relocate. It's a lot of, yeah, commitment. So take research as a main commitment and try to finish as much as you can.
Yeah, I'll do my best. Yeah.
All right, very good.
Alright,
I know,
thank you so much.
thank you. It looks like Richard had to go, but Stas, let me get your update.
Yeah. So a few things to update on. Let's see, where should I start? I'll start with the quicker stuff. So Hassan mentioned Tide to me and I looked into it. It's definitely something we can do. There is one nuance. The format in which Tide takes the RNA-seq data is very specific. So for example, it needs the RNA-seq data to be normalized against an adjacent normal tissue.
Hmm,
And if it's not, the actual, the way they have it set it up, it just won't take it as input.
hmm,
You know, obviously we could probably email them and try and work something out. I think with Tempus, we'll probably have an adjacent normal sample for every patient.
mhm,
Yesterday I was trying to do this with TCGA data, and the
uh-huh.
problem is there isn't an adjacent normal sample for every tumor sample in TCGA. There's
Oh,
actually very little normal tissue.
oh, really?
Um, and so maybe in their pipeline, they've included that data and processed it, but the format that's available, it doesn't work with Tide. So I'm trying to look into maybe, at least for just to test this, using finding some kind of general normal squamous cell tissue that I could use to normalize. I know that probably carries some batch effects and some errors, but it will at the very
Doesn't
least
it
let me.
have to be adjacent tissue?
No, doesn't
It
have to be. No, it could be blood. I'm
Hmm,
assuming. Actually, no, sorry, I'm mistaken. It does because it's transcriptome.
hmm,
You want to remove the transcriptome of the normal tissue or adjust for it so that what we're seeing is tumor. In my mind, it shouldn't really make a difference if you're comparing two tumor samples.
So I would say, let's say you can have background of COPD, and your adjacent tissue will have a lot of COPD
Right, it's a good point.
genes that are upregulated. But that's not tumor. So
Right.
tumor versus adjacent tissue is, I think, is still important. Same thing with Mantis or the MSI high algorithm. They need the adjacent tissue for that algorithm. So anyway, maybe, CC me, ask Tempus, hey, we want to run this with your RNA-seq. Is your normal adjacent tissue available?
Yeah, right. And so that's kind of what I've been trying to investigate. It's not very clear from I'm looking at the S3 bucket right now. It's not very clear from the S3 bucket
Yeah.
how normal factors into it. But
Or they're, I don't know if they did that, the RNA-seq has already normalized it.
right.
Yeah. If it's already normalized, just use it as if it's normalized data.
Yeah, which I've been trying to do because I know the TCGA data is already normalized, and when I try to do that, it gets up. So with any normalized data set for RNA expression, there will be some values that will be positive because they're overexpressed relative to whatever the control is, and negative values which are underexpressed. The
Right.
problem is if you input data that has too many positive values, so if there's more than 80% of the positive of the values are positive, it says, sorry, I can't run this, you didn't normalize. And maybe you did normalize, but they just have this, this parameter set, and maybe I can email them and see if there's some way around it and work with them that way. But that's kind of the issue here. Something else I noticed, it seems like it hasn't really been updated since 2020. So I don't know if there's a more updated tool or something similar that's actively being worked on to try just
Yeah. So yeah, it would be always good to email the corresponding author, like, how did you guys do this? And
yeah.
they did not publish anything from TCGA? Did they, the group,
I don't remember. Let me see.
so look at
Um,
their publications. A lot of group try TCGA at least once, so if they did it, they should have described it in their methods, how they did it. But anyway, you
Oh,
could,
I just went to their website. So like they have a website hosted at
Uh-huh.
Harvard that hosts the tool.
uh,
I didn't actually look at the paper. So maybe I should start with that,
yeah,
go from there. But anyway, that's because I know that's something that you've been talking about this past week. If there's other
signatures.
yeah, other signatures or similar methods, send them my
Yeah,
way, and
yeah,
I can see, I can find the one that takes takes the least time to prep the data for.
Yeah, and then don't forget Tempest, they have their own website. So if you go, so you guys have my Tempest portal ID and password, you guys can, and then when you go to the research section, and they said, and in the research section, there's
Oh, I
a
didn't know that.
signature. There's a
Okay.
signature, they say inflamed.
Oh.
They have their own signature, and they should have their reference, like, like what algorithm did they use for that. And that's another, we could publish that and compare with whatever we've tried to do, with their algorithm. They have, even checkpoint expression, PDL1, everything is HLA, MHC,
Yeah.
everything is there. They describe all
Wow.
that for us. They have algorithm built in. It's automatically populated, but because it's research, they don't give that to patient.
Right.
That in the portal for doctors to study. So explore that as well and utilize that. Because the whole point is, how do these signatures, because a lot of people are doing mRNA based signature, and we don't have to reinvent the wheel. Or
Yeah.
I think our biggest contribution is, how does heterogeneity play a role with all these signatures.
Right.
So, in that regard, and so that's one news for you, is we now have access to whole genome sequencing.
But only for probably a small subset of patients,
So
right?
not our own patients.
Oh, okay.
We don't, our patients didn't do a whole, but we have like like more than 1,000 patients sequence
Wow.
data and their outcome, and through a company who does whole genome sequencing, like as default. That's their service.
Oh, wow. So which company is it?
This is called Genome Insight. They're based in
Interesting.
San Diego, and they are. I'm going to start ordering their service very soon, because they're gonna go commercial in April or something.
Wow.
But they, as part of research, they've gathered like 1,000 patient samples. So we have that and the patient outcome, and then we kind of verbally decided to work together. So we could kind of start reading, because if you think about the exome splicing and all the things that can happen,
Yeah.
with whole genome, and the copy number is gonna be more accurate, and we'll have better understanding of clonality for sure, with the whole genome. And they don't have blood yet, but they will be doing it soon. But the thing is, we should look into how doing whole genome will be more beneficial than doing exome, or targeted panel is limited in itself. We're not looking at other genes. It's a targeted panel of 300 to 500, 600 genes. So we're missing what's happening in other genes. So we don't know what's actually happening. So if the clonality changes in a genome, but that's not exome, but then exome is just like a one snapshot. But some things happen in non-exome, right? And in enhancer, whatever that is, and we're not picking it up by any means. We don't do whole genome. So start reading about it. It's a big project, but I think we'll have really nice collaboration. They do have, bioinformatics people, so we can have them run. I mean, it's a lot of data because it's whole genome.
Yeah,
But at the same time, they're not doing it in the same depth as they do as targeted panel.
right.
But start reading like heterogeneity and whole genome, and we're gonna do some nice project with them soon. So I'll definitely get you involved, but let's
Yeah,
get prepared.
That'd
for
be
that.
great. Do you know how they're making it affordable? Are they
Yeah.
just running, 100 genomes on a giant
So
Illumina
yeah,
sequencer?
there's now breakthrough in technology, algorithm as well as some like regions like they use the way they use it, and the patents are expired. Now,
Oh,
yeah, a lot of so there's almost like it's becoming much more cheaper to do whole genome now.
right.
It's kind of the right timing to start that business.
Because I know some of the newer, larger Illumina sequencers, they can do a full genome.
Yeah.
I think in 30 hours, but you have to run 10 genomes or 20 genomes. There's some, in order to make it affordable, you have to run many genomes at the same time.
Right.
So that's the only thing I've heard of. I'm looking at Genome Insights website. I haven't found how they're doing the sequencing, but I'll keep looking. Thank you for letting me know.
No problem. No problem.
Um,
Liam, on our next PC invite Stas, so that I don't know if you're gonna be in class, but just prepare for it.
Yeah,
Yeah.
I might be. I'm starting surgery in March, so I'm going to be very busy. Unfortunately,
I know.
but I'll
Yeah.
be free in May.
Yeah. Okay.
Yeah,
What
so
your manuscript. What happened to
so
it?
I'm writing it. I have to be honest, I'm really struggling to write the discussion.
Uh-huh.
My like urge is to talk about the data and do that, and I have to stop myself. And so I've written a couple paragraphs, and I just want to get your take and then
Sure.
work off of your feedback. I will
Yeah.
share the link. Okay, I sent it.
Okay, I will share.
So
Okay.
if you just click on the discussion, so I just added this outline just so it's easier to navigate. That's kind of the new couple paragraphs I wrote, trying to write it in the way that you suggested. What's below is my old discussion that
Is this the first two paragraph?
yeah, I'm sorry. I'll write a paragraph and then I'll delete it and write a new. It's just I don't know why this has been so difficult for me, but I feel like if I get your feedback on this, I can keep writing in this style.
Yeah.
And you said start with the punchline. That's what I'm starting with, and then trying to tell a story.
So early state doesn't have to be capital.
Okay.
Furthermore, this finding is intensified. Intensified. I think intensify is a strong word. You could say more evident.
Okay. Yeah.
Best of knowledge. This is the first instance which such ship has been observed. It sounds a little colloquial when you say
Yeah.
I would say has not shown relationship. I think this say it more like specifically
Okay.
is enough. So I don't think you need this, but
Oh, so just get rid of the sentence.
right. And this
Okay.
yeah.
Because it's,
This is we report, or you can support this sentence. This is a short sentence, use of the
Right.
report for the first time that IHC exhibits, right? I mean, like in early
Yeah.
stage, no one looked at
Like,
you know, what ITH does, right? I mean,
yeah, well, there's like, there's all these, there are all these papers out there that will look at ITH and gene expression, ITH, and single, you know, whether it's bulk or single cell RNA-seq. They'll look at protein expression, but very few papers actually say, okay, well, what's the real outcome on clinical progression? There's a lot of these like theoretical scientific papers that say, oh, we've created this framework, or we've given information for people to investigate, but there's nothing like this where it says high ITH outcome. Maybe
Mhm.
there's one with math, but nothing with, there's very few with PyClone that actually show anything.
right. So yeah, really showing differential clinical outcome, you know? So I think this is good enough. Yeah, so this is the punchline here.
Yeah.
Okay.
And then and then next I'm trying to kind of weave into, you know, we saw this outcome or this relationship, and now we want to investigate drivers, we want to investigate TME, and try and make sense of it. And so this first bit is about, this next paragraph is about the drivers, and I don't know.
Consensus understanding is that IP reasonable proxy for potential of a tumor to harbor resist treatment therapy, treatment resistant mutations.
You know, ideally this is something that's, you know, in the introduction, and it is, but I wanted to kind of call back to it to just remind that, hey, this is how it's used right now. And I found a lot of papers kind of using ITH clinically to really investigate these EGFR resistant mutants post-treatment. And so that's kind of what I go on to talk about. But
So in at least adeno, where EGFR mutation is more prevalent, high ITH was linked with poor outcome. But in squamous, we don't know. There are not many
Right.
drivers. So
that's like the logic I'm trying to use not the logic, but the thread I'm trying to use to write the story.
yeah. This is not a relevant information. You can get rid of it because
Okay.
this is a breast cancer argument and it's not relevant for our paper.
Okay. So then instead I can say there are no approved inhibitors that target, you know, these studied driver genes.
You don't have to write whether it's approved or not. That's
Okay.
not the point of discussion. The point of discussion is there's less, less driver genes in squamous, and does that impact differential outcome? Like adeno and squamous are opposite.
Right.
Why is that? Maybe because of driver gene frequency difference.
Yeah.
In our work, high ITH was associated with improved survival in early stage low TMB core to put it.
And then the next paragraph was going to talk about the way that the different cell types are expressed, where they're, and you know, which ones are enriched, which ones are depleted in these respective groups.
So I think it has to be reader friendly.
Right.
I think because in discussion people want to understand why I guess in introduction we kind of did that. Why are we interested in low TMB? why are we interested in comparing ITH high?
Yeah.
Here, start saying that, we've seen that impact of ITH on clinical outcome was different, squamous versus adeno. And, we were interested in understanding why. And we focus on the difference in prevalence of driver mutations between
Okay.
histologies. And then say what we found, in adeno, low ITH, what high IT what was better prognosis.
Yeah.
High ITH was poor prognosis, and that's probably because of potential resistant mechanism co-residing along with the driver mutation. While low ITH may mean solely driver mutations present. In squamous cell, there may be a totally different biology playing a role there. And because of the low frequency of driver genes, and one argument that I don't think we had good hypothesis to explain.
Right. I did
So-
have, right, I did have one idea that, although we don't really have data for it, maybe we can generate some that could be interesting. Is last year we read a paper where they looked at clone to clone competition. And in that paper, they showed that if they expose the tumor to treatment, the clonal kind of profile changes. Then when you remove the treatment, it changes again. And so the thought I was having is, maybe in adeno, or rather, sorry, in squamous, where you have high heterogeneity and a better outcome, you have more clone competition, preventing the development of more aggressive clones, or preventing aggressive clones from having access to resources, or some relationship between this high number of clones leading to kind of a more stable overall state of the tumor, because they're competing with each other. And that's
Yeah.
not something I've seen discussed elsewhere.
Right. I think it's hypothesis. I
Right.
don't think we need to write it out without, like a firm
Yeah.
clinical evidence, but it's a good thought. But, what about adeno? Like, how is adeno that different than the competition theory doesn't apply? It's still driver mutation
Right.
is clone, so it's hard to, you have to say what is the core difference between adeno and squamous and go from there. If you remember, I think Haesung did nice literature review on that cell paper on the radiation induced ITH experiment, and in that paper it was still the high ITH, high heterogeneity was linked with worse response to immunotherapy or survival in mouse experiment. So ITH with favorable survival outcome is sort of foreign to the world. But that's what's happening. And then maybe
Right.
if the fact that we selected for low TMB, so that's why last time I said we need to see what's happening in high TMB and show both.
Yeah.
And the answer was it's more pronounced in low TMB, but in high TMB, did you see the similar trend, or what was I forgot what you saw.
So the high TMB, I put that in.
Is it in this
It's-
file or a different file?
Yeah, no, it is. It's in the supplemental. It's the second figure.
Oh, but not here.
It's in the supplemental. So if you click results and then go to supplement, oh, yeah, and then scroll down to supplemental figures, and it's the second figure.
This one,
Yeah.
high TMB, high TMB. Oh, they're all high TMB. Oh, this is low TMB. Okay. So we'll see low TMB first. In low TMB, squamous.
These are different, sorry, these are different cutoffs. These are the not statistically significant cutoffs. So that we're not like, if you want to look at the low TMB, that would be figure 1. Sorry, figure 2.
This one?
Yeah, so
Oh, it's supplemental. Oh, yeah.
sorry, there's a lot.
Oh, yeah, now I see. So this is supplemental and figure.
And in this figure, I think last time you gave me a recommendation of combining some of my figures. So this figure contains ITH only, and then ITH with low TMB.
Okay, so this one is combined.
Yeah, so the figure has what ITH on its own does to patient stratification and outcome,
Right.
and then what it does with low TMB to kind of go along with the punchline of ITH has this relationship intensified in low.
Yeah, intensified in low. And
Although intensified is, like you said, a little strong.
yeah,
And then in high, what's interesting is squamous cell doesn't have anything that's significant. There's also smaller sample sizes, but I think in adeno we see a similar relationship.
Yeah, so there is a trend, maybe,
Right.
In squamous, and it's still the opposite. At least the direction is same. For
Yeah,
adeno, high ITH is worse. For squamous, high
Yeah.
ITH is better. But it's more pronounced in low TMB. So I think, just so for your discussion, I think just go into that logic. Why, we saw this difference. Why is it? And this is what we think may be contributory.
so just driver genes. So that's kind of our
And squamous,
hypothesis.
yeah, it's low, and there may be a different biology, and just stop there. Don't try to explain.
Right. Like the idea that molecularly there's this fundamental difference. There's these highly aggressive drivers in adeno and none in squamous. And
Mhm.
so that's the explanation. But I feel like won't the reviewer want some sort of, well, who cares if you talked about this difference, but how does this difference relate to ITH? Like what is
Mhm.
what if this is just a coincidental finding? That, I mean, there's other stuff that I found that has a differential relationship. So there's a lot of scores that I found, immune scores, stromal scores, that, and a few other things that have this opposite relationship. There's
Right.
also an opposite relationship when it comes to the impact of certain immune cell types on
Right.
outcome. So for squamous one cell, enriched one cell type, I forget which one, maybe it's macrophages or Th2s, I don't remember, but it's enriched in squamous and that results in a better outcome. But when it's enriched in adeno, it has no effect. Stuff like that.
Yeah, so you can vaguely say the immune microenvironment of these two histologies are very different, and then in immune microenvironment and how ITH and immune microenvironment interacts may be different, affecting different clinical outcome. So you can kind of vaguely describe that.
Yeah.
Maybe the immune environment is different.
I mean, I can say that we saw a difference in the immune microenvironment, and then here's some papers in which they also see a difference.
Yeah. Those papers haven't linked that to different
ITH.
clinical outcome, but we did.
Right.
Okay. So yeah. I
Okay,
think
this is helpful,
rewrite
yeah.
that. Yeah.
And then as far as the length of the discussion, I'm getting the sense that it should be on the shorter side. It should be very much to the point and on the shorter side compared to, that's my old stuff,
Yeah,
compared to the results and introduction sections.
right. Yeah, so discussion has to discuss anything that's, related to our topic.
Yeah,
Any previous publication and worthwhile mentioning, like, and then how does that support or not support what we saw?
right,
And then in the summary, like this is kind of first step, understand the impact of ITH in different settings, in different histology, different TMB setting may be important. Like we can't just say ITH can be
right,
the same impact. The impact of ITH may be different according to histology, number one, or according to TMB, number two, or according to immune microenvironment, number three. And this is kind of the first baby step to
yeah,
understand that. So that's kind of our,
right.
wrap up here. Okay, very good.
Okay.
I know it's hard to write it
Yeah,
out.
this has been tough. Yeah, this
Yeah,
paper is
I mean-
just,
And analysis is one thing, and making sense that people making sense out of it, and like
Yeah.
writing a good piece is a totally different thing. But the more you think and try, you grow. So this is
Yeah.
the way to do it.
I've definitely learned a lot in the process. So
Okay.
it's been good. Thank you.
Good. It's us. And yeah, still work on like getting the raw data from Tempest and like
Yeah,
not Tempest, but the Guardant. And we need to know if they have the whole 500 gene, not just the 70. So
well, they don't. It sounds
Oh.
like I email, let me pull up that email. I talked to them last week about this and
they don't.
let's see. She wrote, basically I asked her about larger panels and stuff.
Yeah.
And they wrote, there's been no changes to our panel. We have a 74 gene Guard 360 CDX assay, as well as the Guard 360 LDT assay, which is 83 genes, which includes TMB, which is assessed on the backbone of 500 genes. So it sounds like they use 500 genes to assess TMB,
Right.
but for specific, but for some reason they only use 83 for their gene assays.
Okay. So they need to know in that 500 backbone of 500 genes whether they have mutation and what their copy number is
Right,
in order to calculate TMB.
right,
So the next email CC me is going to be, we understand that you have, mutation information
yeah,
of that 500 gene in order to calculate TMB. So, can we request that 500 gene mutation data? The depth of sequencing, those don't matter. We
Right.
need that 500 gene data to be able to do our heterogeneity research. We know you guys ran it, and otherwise you wouldn't be able to
Right.
calculate TMB. So what is the best way to have access to that?
okay,
That's
Yeah.
our next question.
all right. I'll reply to that email after the meeting.
Yeah. And CC me and Liam.
Yeah, yep.
Yeah.
You guys are on this. You,
Yeah.
Liam, Hassan, and me.
Okay.
And then I think that last time we talked about Oncocyte or the dermal IO
Right.
is, I think, yeah. So I haven't heard anything back from them. I don't know if Liam has, but it's unclear whether, it's still unclear whether the Tempest RNA-seq data will fit into their pipeline, given that they don't cover the same genes.
Right.
So we'll
right.
see.
Right. So, yeah, I understand. So, did they respond to that email? Like, were they willing to do it without?
They told us they'd get back to us, but I don't know if we reached back out to check in with them.
Yeah, just send another email to you guys, because we want to work with you, but I know you have number of gene, but I don't know. I would be interested in checking out like what happens if, one gene is missing, like, it's a major compromise,
They told us it would compromise it because that's their like secret sauce. They use this
Yeah.
combination of cold genes and hot genes, and if we only have the hot genes or a fraction of each of those, it's not enough.
Mhm.
Um,
But we're
Yeah,
missing only a few, right? Like it's.
they told us they'd get back to us and tell us which genes were missing and how many, but there
right.
hasn't been any word from them.
Yeah, so just send that follow-up email, CC me and see what they say, like, can you?
Okay.
But in the meantime, yeah, work with Tide team and see if there's any better signature that's been recently published.
And you're specifically interested in signatures that predict IO response
Mhm.
or
Yeah,
okay.
IO response. That's our data. And shown validity in lung cancer. I think the signature may be different from melanoma. There's a melanoma directed signature too.
Yeah.
And that signature has been published too from group in UCLA. Now there was a name to that too, but that's more melanoma based. Okay, so yeah, keep working on
Yeah,
that.
yeah. Okay,
Okay,
I'll send those out today and do some reading.
okay, very good. Okay, so Jisang, do you want to go next? Because we have abstract due today, so.
Right, right. Let me share my screen. Can you see my screen?
Yeah.
Right. So I received from the answer the result from Yuri. You yeah, we tried our best, but there is no significant results. So Yuri said, so we have total, I will correct it. We have 103,
Mhm.
the total sample number, but
Mhm.
Yuri said we should collect 3 to 4 times more patients to
Mm.
overcome these issues. So I'm not sure if we can do the to get a positive result. So I trust him as a statistician. So yeah, so it says so the result shows also the negative results again, and he gave me the R code for the analysis. So so using this code and our data, we can run the analysis
Mm.
by changing the variables, so we can get the results we want.
Mm.
So these are all variables we gathered. So if you want any specific variables that you want to see, then I'm gonna choose those and run the analysis for the results.
Mm. Okay.
So could you select?
So he hasn't run any of it?
So he sent the sample only response. Let me check. So like this. So he run the response,
Hmm.
and the results show like these, and you can see the sensitivity, specificity, PPV, and PPV like that. So I guess we can get these kind of results when you run the code.
Can you show me the table go up? Yeah, this one. Can you explain what this is?
So this I can only interpret the results of sensitivity and specificity like that, not about this table. But I can guess this is the standard uh, and so this is I'm sorry, I don't know about this table exactly.
Hmm.
So
So what about
So-
responders and non-responders? Did you guys do it like 2A? This seems like a 3 PDPRSD.
per variables, it can show 2x2 or 3x3 table.
Mhm.
So if the variable has yes or no, then it can show 2x2 or like
So yeah, so durable response, yes, no. Durable disease control, yes, no. So did Yuri use that variable
oh
to?
no, he run this response data. So we have no CR, so it shows only PD, SD, and PI. So
Hmm.
it shows 3x3 table.
So I think you need to run
Durable disease,
durable response and durable disease control
sure, of course.
and see what that predictability is.
Right. And any other variables you want to run?
So if we don't show any of so let's say if the AUC is similar to what we found in AACR,
Mhm.
you report. I guess even if it's worse, I think we report. We have to report that radiomics is not a good predictor. It's a negative study.
So I'd like to ask if you have any plan to add more sample like you suggested.
Well, I like to, but we don't have more samples. We have to treat more samples. And I mean, we can reach out to other investigators outside Northwestern and get more data. But this is a,
So I'm a little bit suspicious if we don't have enough sample number to run the
So it's the
study.
clinical study is report with what you have. Even if it's negative, even you've done what you can with your data. And I think so it's like if you've seen small success, see if it's going to be a bigger success with a bigger population.
Mhm.
If you see a negative result, you have to invest your time and money, your everything. You need, so it's an investment. Are you willing to invest more in radiomics? by reaching out to others to collect more data? My simple answer is no. I don't want to reach out. This is the best we've done with the data that we have. Our immunotherapy patients numbers will increase, and we can check it maybe after 2 years. We add more people, and there will be a better algorithm in 2 years. Trust me, the field is growing very fast. So we'll have a better AI engine that will better predict, and we'll have more patients. We can retry that. But, that's why we need Picture Health and Lunit to tell us that, they have investors. People are investing in those companies because they think their algorithms is superior. So we'll give them a chance to prove that with 100 patients. If they can't say, if they also say the sample's too small, it's small. I mean, like, if you can't say anything meaningful with 100 patients, like, I'm not willing to invest 200 more patients. So how I take this is a publicly available. Yuri is academic investigator. Like, he should know what's the best algorithm out there, and he's applying it. It's not making difference. So these companies are saying we have secret sauce. Okay, we're looking at vessel tortuosity, and we're looking at different things that other people are not looking. So we're not sharing the algorithm. Rather, we made a company. Okay, so let's see if your secret sauce works well here, and publish if it really is working well. And if it shows success, I'm willing to get other people participated. But companies will have better motivation than I do because they want to sell and make money. Okay, I'm not there. I'm not doing this to make other companies make more profit. I'm doing this to see, ask scientific questions. And is a really good biomarker for our patients that I want to utilize in clinic. So the answer is, let's look at these variables now. And even if they show negative results, still write up a abstract saying that with publicly available algorithm, we were not able to do well. Like, there's a very weak association. And AACR took it. So I don't know why they took it. They want there to be no hype about radiomics. Maybe that's why they selected us. Maybe ASCO will do the same. We don't know. We report. So beauty of clinical study is, if you do clinical trial, even if it's negative result, you report. Okay.
All right. So.
And you can look at the same things you've looked at AACR. Whether radiomics can predict PD-L1, MSI high, TMB, specific EGFR mutations, smoking. You can look at everything you've collected for a reason. So look at everything and see if you find any meaningful correlation. If you don't find anything, to say you've not seen any meaningful correlation.
So do you want to put all of these if there is no correlation
So in the
at all?
method, you say, we've looked at all these variables in the methods. In the result, you can say, among all those variables, we found no or only one association with one variable. That's how you report it.
So as I put the table in abstract for AACR, so
do the exactly same thing you've done for AACR for
Right.
ASCO.
Okay. I see.
Any questions?
So I just want to ask, so at that time we submit abstract for AACR, we choose some variables, at least a little So they are all mm, uh all were not significant statistically, but we put uh some variables on the table. So in this time, I'd like to ask if you want to put some important data to show on the table.
So in theory, if nothing was significant, you just say not significant, and that's it.
Right.
But if you see like a trend, p-value close to something, and then you can at least describe it. And I don't know if you need table here or not. You can, I think we did table in AACR, so you can try it.
Mhm.
And make one and show me, and I'll give you my comment whether I want the table or not, but.
Okay, I see. Yeah. Right.
And
Mm,
for AACR, did we have 2 abstract or 1 abstract?
only one.
Okay, so just follow that. Yeah,
Okay, all right. Then I will run the analysis by our code and then finish the abstract and table, and I will show you.
so do you think you can do that by the end of today?
Yeah. So Yuri told me kindly how to run the code. So I only need to change this word
Uh-huh.
for another variable, then I can run all of this. So Dr. Myung-woo Nam has R on his computer, so
Mm.
I need his help run the R because I don't have R. So yeah, he will help me to run the test.
Okay,
All right. So
oh
could
yeah,
you
work with Myungwoo.
mhm. Could you choose the category for a ASCO for this radiomics? So I couldn't find any radiomics category or at radiology like that. So
Um. Biologic correlates.
there are more categories that I choose only relevant categories that I felt.
Biologic correlates, not biology. Other IO related topics. Well, I guess we looked at only lung cancer.
Right, non-small cell lung cancer.
Hmm. I think we have to do biologic correlates for metastatic lung.
No, okay. These
Yeah,
ones.
and there's no other category.
Thank you. Um, yeah, AACR poster service deadline is on March 24th. So I will add a figure method like the
Uh-huh, uh-huh.
I just search Google and put the figure, but yeah, I will make it like this.
Yeah, yeah, something like that. Just make it into your own, yeah, version.
Okay, and for yeah, image prior, I have a plan to collect all scan data,
Mhm.
patient name, date of birth, for identifying patients, and date, scan type, and I will add these target lesions and non-target lesions information from your advice, and then request Edwin for DICOM file. And I will also learn and upload to image trial beta version, and run the resist and get the results. So I think within a week I can collect all the scan data with help from other colleagues. Um, so
What is
yeah,
Edwin again?
sorry.
What is Edwin?
So Edwin is who can give us the DICOM file from Epic.
Oh, it's a person in
Yeah,
charge?
right.
Oh, okay. Okay.
So I'll add the information like
Mhm.
on Excel, and we will gather information. And about double lung transplantation, I sorted these UNOS and ISHLT data. So I just uh, make an algorithm for this from UNOS data. I just stored double lung transplantation and exclude the patient with previous malignancy. And then I sorted patients without post-transplantation malignancy and with post-transplantation malignancy. So I already sorted and organized the data and sent Dr. Jian regarding UNOS data, and I'm still on the process of sorting data on ISHLT.
Oh, okay.
So just
Yeah,
I just want to show the number of patients
yeah, there's
and
still a lot.
the output. Yep. So right, so about more than 20,000.
Yeah. So 4,000 out of
Yeah, 4,000.
20,000
Right.
will develop cancer. That's pretty high, but that's okay.
Right. So I just only have 2 weeks left. So I will sign out radio mix and double lung transplantation. So um, so I heard from Dr. Jia. She will be in charge of the radio mix as a key person. So I will give her all the data, and also I will share work to other colleagues, like image trials or picture health unit, and then yeah, double lung transplantation as well.
Yeah, so when you sign out, like make sure Hesong and Liam listens, understands, because they'll be here, longer than Dr. Chian, but when Dr. Chian is here, work with her to wrap up and publish everything that can come out of it. But yeah, so the expectation is, while Dr. Chian is here with us, we finish the Lunic collaboration, Picture Health, whatever we found out from Yuri's lab collaboration, we'll publish. But if anything lingers on, then Liam and Hesong needs to know. So during the sign out process, I want Liam and Hesong to also listen and understand what's going on. Yeah, so that's pretty much it. So the lesson for radiomics, we always talk about data overfitting. There's that concern, but also there's so many factors. What's the machine? What's the contrast, concentration? So many things. Patient has COPD or not. There's so many variables that come to play in image, making that tumor look like what it is. And there's solid tumor nodules, but there's lymphatic growth tumors, which is very hard to say. Is it pneumonia? Is it tumor? We don't know. So there's an observer variability, a lot of it too. So the more the more confounders you have, the less the predictive power you'll see. So it's difficult, but we know it works in other fields. So in the field of immuno-oncology, does it work or not? There's papers saying that it works. There's recent AI paper that shows that was it mammogram? They looked at mammogram and they found, oh no, there's a mammogram paper too, but there's also low-dose CT of chest that just came out. It's called Cybele, is the program they used, and they found out that even long before we found lung cancer, they were able to detect that change in carcinogenesis from the CT scan. So that's easier because you're dealing with normal lung, and any simple change they detect. But we have patients who already have lots of tumor and other pathology, pneumonia, scar, had prior radiation. There's so much going on in the lung. And then you're trying to figure out whether the therapy works or not. So we're asking a very complex question here. So AI may be best suited for early cancer detection through mammography or low-dose CT scan. But to get to the therapeutic decision-making arena, it may be tough. But again, we will continue to report what we find. But the research starts like this, and a lot of times we don't find anything interesting, but we keep doing it, with different algorithm, adding different aspect, and sometimes a breakthrough will come. So staying faithful in the field and kind of follow it up and see what you can do. So it's a humbling process, and I don't want you to think that I put a lot of time into it, I don't see good result, it's disappointing, and I don't want to do research anymore. That's how a lot of students feel. So I want you to know that in most cases, I write a proposal for clinical trial. If I submit 100, only handful, 2 or 3 will get accepted. I write grant to get funding, less than 5%, I'll get funded. But I still try, write it up. If you don't write, things don't happen. So I want you to know that it is the persistency that matters in the end. And if it's a negative, let it be. We don't manipulate data. We have to know what the truth is, where the technology is, and is it ready for prime time? If it's not ready for prime time, don't use it. It's a waste of resource. We have to be the one to be able to say that. And I've met a lot of thought leaders in the field, and a lot of them told me I don't believe in radiomics. I met world leading immunologists. They told me I don't believe in hyperprogression. I don't. So everyone believes in their own thing. Same thing. I met people like my own colleagues, like I don't believe in lung transplantation. I think it's unethical. So they're very strongly opinionated about what their beliefs are. So what we do is go back to the data. What's the best available evidence say? and what is the best available technology out there. Get those people to become your collaborators and analyze the data in the best way you can and say, hey, this is my conclusion. I don't think we're ready for prime time, or I think we have a promising future ahead. So that's how you contribute in the field. So but the good thing is, during this process, the company you're working with can go bankrupt, and everything all your time and energy you put into may be of no value. So those things have happened to me many times. The company disappeared and my colleague, disappeared. A PhD student, worked for that professor 7 years, and the professor gets stroke and go to a nursing home. You have to find another mentor, you start all over. Things will happen, but what does not change is the rigor, the scientific rigor, and all the skill set you learn along the way you think scientifically, and the persistence that does not go away. So I want you guys to stay strong. It might be exhausting, you may feel it sometimes, but that's how we do science, and it will always pay off in the end, in a more intangible way. It doesn't have to be in the form of making more money or becoming more famous. No, it's in the understanding the truth. If the truth is negative correlation, then let it be. And but we have to make sure, did I explore all the options? Was my methodology rigorous enough? So, okay, thank you. Myungwoo, I know you have abstract due today.
Yes, so for the, what was it, the glycoproteins. So yesterday, Dr. Isa and me ran the statistics for IRAE. And I'll just briefly show you the results.
So for those of you who does not know Myungwoo, you're a second-year resident
Yes,
in internal medicine in what hospital?
and Lincoln Hospital in Bronx, New York.
Lincoln Hospital in Bronx, and he is interested in research, and he's been in our lab before. He chose to come back. This is your vacation?
Yes,
How many weeks?
4 weeks.
So he chose to spend 4 weeks of his vacation in my lab. So on the day of arrival, we told him tomorrow's the deadline and work on the project. And this is the glycosylation pattern of serum proteins, and seeing if that predicts survival outcome of immunotherapy treated patients and immune related side effects.
So it was a little bit crude. So we ran the t-test, as you mentioned,
Yeah.
for using the glycoprotein.
Uh-huh.
And surprisingly, there was only 22 glycopeptides that were significantly different between two groups.
Okay. So you
One-
put 22 on the right y-axis.
No, so this is for the X, this bottom part, this is the 22 glycopeptides, and this is how they cluster using the R. This is what Dr. Isel did.
But this is not
So-
22. This is
So in the bottom, this is supposed to be 22. I think the letter is large, but I think this one column is 21 each.
oh, it's 22.
Yeah, I think that's 22.
Oh.
So what happened was, so between patients with and without IRAEs, 22 glycoproteins were different.
Mhm.
And we used those 22 glycoproteins to cluster patients into two groups, mainly. First, we,
Yellow color looks like a real outlier. I don't know what happened to the sample.
these
What do you
ones,
think? The yellow one.
this one?
Yeah.
Yeah, I'm not sure.
Yeah. Okay. Anyway.
So yeah, we ran it, but we weren't really sure how to interpret this, but we just used to get a cluster, group
Okay.
them into two. So what happened was, initially we grouped them into just two groups,
Mhm.
and if you can see, this is the green part.
Yeah.
So first, when we grouped in just two, there was this green part versus the blue plus the red.
Yeah.
They have clustered it, as you see in this diagram. This makes sense, but this was only 4 patients, the green part. So what we did was we grouped them into 3, which clustered them into 3 groups, and we combined the green plus the blue and
Yeah.
compared it to red.
Yeah.
And we divided the groups into 4 and then compared red plus the others like this, both of them.
Okay.
So, and then we chose the left one because this was a little bit more significant, and also the number of patients in each group like made more sense just to us, because this was about 37-ish versus this one was like 13. Here
Okay.
it was like 27 versus 20, which is like about half.
Okay.
And we even tried to see if this cluster can predict IRAE greater than 3, which didn't. And so we just ran the not the PFS, this one, the IRAE, development of IRAE.
Mm-hmm.
So 1 is the red one, the red cluster group. 2 is the rest, which showed significant difference. You can see there is like a more predictive value in the cluster 1 group.
So go back to that graph, heat
Oh.
map. So can you make, because it looks, to me, it looks all red and some orange in the heat map. Are you, I don't know if you are, it's yellow to red.
Mhm.
I don't know if you can make it, different colors, red. Because
So I
I don't see much separation between.
Yeah, I think this part is the separation they made based on this. So this glycan maybe has some more value, like compared to other glycans.
Yeah.
Yeah, but I'm not sure if there's significance. It's only like 1 or 2, maybe.
Yeah. Yeah, I would have thought some color, gradation or
Mhm.
something here, but it's not like all
Very subtle. Yeah.
very subtle. Anyway, the graph you showed me said red versus,
Red versus blue plus green.
okay.
So in
The reason you didn't do the right side was why?
we ran both, but
Uh-huh.
left was a little bit more so for the like a PFS or the development study, we saw the both, where did it go? Both like log rank test and the other method. For the left one, both were significant. For the right one, only one was significant. That was one reason.
Okay,
Yeah.
okay.
And when we ran with OS and PFS using this 22 genes cluster, there was no significant difference. So we could argue that this is more specific to IRAEs, maybe. Oh,
Yeah. But OS was no difference too?
no difference. PFS was no difference. Only the IRAE
Okay.
showed
That's
difference.
good.
And just a second. So for description, there are not many to describe, but if you see here, this is the p-value. So median survival by the program, the group 1, which showed the the worst worst result, had undefined median survival, whereas the better cohort showed 91 days. So if you have like plenty of time, group 1 does like worse, but the time to development was like the faster in 2, which I don't know how to interpret. And the other thing was this hazard ratio is actually not significant. It includes the 1.
Uh-huh.
So I don't know if I'll have to like write it here, for example. Like, should I just mention the p-value or should I just mention the hazard ratio? And this median was just done manually in the Excel spreadsheet.
So median is
Yeah.
time to,
development of the first IRA.
so yeah, I think you should write that.
Okay.
So show me the hazard ratio again in that analysis.
So here it just says the group was 1 and 2, but here it just says B over A. So I just figured B is the worst one.
Yeah.
Yeah.
What is the p-value?
They don't have the p-value for hazard ratio actually.
There's gotta be a way for you to calculate that. Maybe just use the log rank. Yeah, log rank is
Mhm.
fine.
So this one and this one. I'll just write this.
Yeah, just write those two.
And the median is just from manual Excel spread, not this one.
Yeah. Uh-huh. I do want you to still write when signature derived from
Mhm.
better response was utilized, then we were able to see the trend, but not significant. The
Oh.
one Dr. Isers did, you can still write that.
Oh, you mean the previous, the
Yeah.
one? Okay, yeah. I just said it. Would you like to go over briefly this one? I just finished up everything. Just result part is the one that's missing for now.
Okay, the title, potential role of serum glycoproteomics in predicting immune-related adverse events in treated with immunotherapy. So write out non-small cell lung cancer and then put NSCLC in parentheses.
Just start over.
And you don't need to put reference introduction number 1.
Oh, okay.
Oh, in the method, because in the title you put so what I meant is non-small cell lung cancer, put that in the parenthesis in the title too.
Okay. But because sometimes they don't like allow the thing.
The word limit, it's okay.
No, not the word limit, the abbreviation. And title.
Oh, yeah, and then in the method you
Okay.
can put NSCLC because it's the title. That's usually what I do. Is it 40%?
This paper like 30s to 40, some cancer 40 from something.
Yeah. What about our patient? What was the percentage?
I think let me see if I have it here. 43%.
Okay. So we have to write that in our
Yep.
results.
I'm gonna write it over here.
Yeah. So don't put reference here.
Oh, okay.
You don't need a reference for abstracts. In predicting in you don't say in predicting AIs in patients with non-small cell lung cancer receiving immunotherapy. You don't need to say our hospital. I mean, that's pretty
Oh,
sure.
this one.
Yeah.
Yeah, I mean, I
Don't
thought
have
this
to
is,
put Northwestern Memorial Hospital. So, and then 47 patients were, so consecutive 47 patients. So it's not like we handpicked them.
Mm.
We did chart review and found out from the, time period. So I don't know. So in the, you can leave it as in the method like that, but for the abstract, you can do, for the manuscript method, you have to write how you got 47, like from this year to this year, we and the eligibility criteria was this, and so consecutive is important. You don't have to write Northwestern Memorial Hospital. The people can look at the affiliation. Treated with a treatment regimen, including. So 22, one was non-glycosylated. So how did you put them all together as like abundance?
so actually the abundance in the raw data set, the abundance raw data set had all these genes. And when we were going through the 22 gene, not the genes, the 22 glycoproteins in the Excel sheet,
Mhm.
we found that one was just like, didn't say glycosylation. So we figured it might be just non-glycosylated, but to think of it, like, yeah, it's weird. It's not then abundance. It's if it's not glycosylated, it shouldn't be there.
Right. So if we don't know for sure,
Mhm. Should we just leave this part out, like just 22 biomarkers?
yeah, but when you say biomarkers, what are you referring to?
Oh, glycopeptides, then maybe I'll just go with,
Yeah. Oh, well, you've been using GP, so if you,
yeah, oh,
okay. So when you say ICI plus chemotherapy, ICI alone. Potentially be a more specific biomarker. I think this is overfetch this
Mm.
because this is how you develop it,
Yeah.
like develop. So when you say GPs can be more like, it's just a general any glycopeptide, but what we're saying that a difference in patterns of patient clusters defined by distinct glycoproteomic signature. That's the better way. distinct glycoproteomics signature. Showed, or you can say predicted, not showed, predicted
Oh.
development of and stop there, just period. This signature did not predict survival outcome, or did not predict response to therapy or survival. Well, I guess that should not be conclusion, write that out in the result. It does not have to be emphasized in conclusion.
Oh, this one. Okay. So I guess this should be enough, right? Or do you want like this?
So this is vague when you say no, there was,
Oh, okay.
so when you say, right, yeah, so this is, so yeah, use the term, well, yeah, so the signature predicted development of IRA. It did not, okay. Okay, validation of our model in the larger quarter warranted for further. Not for further study is warranted, period.
Oh, further study is.
Uh,
Is it okay to keep this sentence, or should I just erase this?
Erase it because it doesn't mean much. So with larger cohort study, or further, to take out or further.
Oh, larger cohort.
So maybe in conclusion, our result for the first time, our results suggest for the first time no, the very difference in patterns of
Oh, okay.
or you can say our results suggest. So we report for the first time. Okay. That's good.
Yes, I'll just refine it. Yes, and that's it for now.
Okay. What happened to the other abstract?
So that one, Dr. Lisa Kim will change a little bit because most of it is written, and then she said she'll, I think, submit it later today.
Okay,
Yep, I'll follow with her.
so she said she forgot to, I followed up with her this morning and she said she forgot to do subgroup analysis. So remember I
Oh.
said do chemo immunotherapy only and show predictive um trend in that cohort. She said she didn't do it, so she said she'll send you the raw data
Oh,
for
okay. Okay.
subgroup analysis. So just add that subgroup analysis and once you written the final abstract for both abstract,
Mhm.
print it out and bring it so that I can have a final look at it before I go home today. Okay.
Yes, sounds good.
Same thing, Chisang, when you finish your abstract, print it out so that I can edit the final sentences together. Okay. Okay, anything else, Myungwoo?
No.
Okay. Okay, we'll go to Jian. Dr. Jian.
Let me share the screen.
Oh, it's been, so let's take a bathroom break. We've been
Okay.
doing it for 3 hours. Okay. I'll be back in 10 minutes. Yeah. Okay, let's resume. Okay, Dr. Jian.
Yes. Okay. So I'm gonna start with the project double lung transplantation. And original article and review article both are submitted to Journal of Heart and Lung Transplantation. But I don't know why, but the original article already they're made in their decision. Maybe it could be rejection because it's been only 3 days
Decision in process.
since I submitted. Yes, yes. So I think I have to decide the next journal at this point.
This is
Yeah. Anyway, how about the next journal?
American Journal. of
Yeah, I know.
Yeah, American Journal of Medicine. That's good.
No. Yeah.
Yeah.
Um,
um,
yeah, still transplantation journal is
Better. Yeah, I think it will have impact.
proper for this article.
Hmm.
Right.
So transplant reviews.
Yeah.
Is it
And there's journal.
a review only journal or they take
Mhm.
original article?
Yeah. Excuse me. Can you say again?
No. I said it's the transplant reviews review only journal or they take original
Review.
article? Review only?
Oh, I don't know, but the one I have to submit again is original article, so there will be no problem.
Yeah, so but I think transplant journal is good and just following this. Yeah, as long as you think those are good, I'm okay with this order. Yeah.
All right, thank you. And third journal, Dr. J gave me the data last Friday, and during weekend, and yesterday, Dr. Andrew and Casey and Hanson helped me a lot in analyzing. So we already analyzed. I think I can make an abstract for ASCO today because there are so many data research also. Yeah. So when you look at this graph, this is swimmer plot graph. Skin cancer is the most common post-transplant malignancy,
Hmm.
especially squamous cell origin. And lung cancer was a fourth common
Hmm.
malignancy. And here, well, briefly, I analyzed just the overall survivor.
Uh-huh.
But interestingly, the cohort who had post-transplant malignancy, they showed a better overall survival significantly.
Oh, that's interesting.
Yes, interesting. Yeah, yes, I analyzed it 3 times, but
Uh,
the results were saying I think I can make an abstract.
So do you know, oh. So, for both you and Chisam, do you know the graft failure, the rejection, graft rejection, data post-transplant?
Yes, we have.
So you know, and we could show reverse relationship between graft rejection and post-transplant malignancy. This is a really interesting concept that we've been dealing with, and this has been written in Dr. Eugene Oh's review paper as well. So what we're looking at is same thing with transfusion. If you get a lot of transfusion, what happens? Your body becomes immune tolerant because you're exposed to so many different people's blood. You become very tolerant to foreign stimulation. So let's say you get organ transplant, but you had and then let's say a group had no prior blood transfusion, another group had many blood transfusion before. The group with prior blood transfusion has less graft rejection because their body has been very immune tolerant. So the graft survival is much higher. But the cancer can survive. Graft rejection and cancer rejection are same phenomena in the human body, okay? It's a foreign thing. One came from surgery from outside. One internal genetic mutation and becomes a foreign tumor within our body. So our immune system has to reject both. So if this will be a very interesting paper if Dr. Jian can show graft failure and post-transplant malignancy are opposite phenomena.
Yeah, okay, I'll take a look.
Yeah, so the reason that so this is my hypothesis, the reason why people who develop cancer live longer is they don't develop a graft rejection as high as the other group.
Okay. That's
And survival is often limited by graft failure. And it
Alright.
happens with, rejection. Immune system, so immune tolerance is important if you have organ transplant or if you have lupus, inflammatory bowel disease, autoimmune disease. You need to have a tolerant immune system. But tolerant immune system is bad if you're dealing with cancer. You need to reject all cancer cells. So that's why immune system is a double-edged sword. Okay, so I want you to understand this, but I think I love this graph because we're kind of asking that question here.
okay.
Okay.
So, Andrew, can you show the each malignancy graphs? Because he analyzed it yesterday a lot.
I'm gonna share it.
Yeah.
그래서 검소고?
I can listen you.
So this is a graph for post-transplant malignancy, total 27 graphs made by Casey and me. 4 to 8 to 12 to 16 to 20. These are 24. This is through 27. So there are total 27 graphs ready. Any feedbacks you would like to give it to me? Anything I should change?
Well, I tried to see the trends in each malignancy. When interestingly, like a brain tumor,
Yeah, show me.
it occurs after 3 years.
Yeah,
show me.
show me the graphs again. So lymphoma tend to happen mostly first year. Skin, second, third year. Lung, first year. Skin kind of lingers on. Next, colorectal. Prostate. Skin. So what, this is melanoma only skin?
Yes,
Okay. The other one was non-melanoma skin, right? So just make sure you
uh,
non-melanoma skin.
those were squamous cell origin
Uh-huh.
and also basal cell origin. So
Oh.
the skin cancer classified as a 3 groups.
Oh, okay. Bladder and go down. Breast, kidney, pancreas, esophageal. Okay. Tongue, genital, thyroid. So I think go to the first one. The trend is pretty clear. There's a lot of lymphoma, 571. Lot of skin. And the basal. So because this is a lung transplant, so the first year recurrence may be from, I don't know, contamination, or maybe from the immediate, some cancer cell in the body coming back. So this is more I think in the transplant world, they use immediate recurrence and delayed recurrence. So they have a definition. I think they use 1 year or 6 months as a cutoff. So see what the transplant world uses as a timeline. So I would say first 1 year, what is kind of cross-sectional first year.
Okay.
Like maybe a pie chart of what is the
Mhm.
first year and the first and then and then the second, third year, and then the
Mhm.
following. How does the, let's say, pie chart distribution change during that time?
Mhm.
So that's good. And so lung cancer has to be the focus here as well, because the reviewers will ask, why are you limiting to double lung transplant? Our answer is now double lung transplant is more, I guess, on the increase, that we're doing
Yes.
more double lung transplant, and that we need to know what's unique about, what are the unique opportunities, challenges in double lung transplant. So that's why we're doing it, and we want to know, what is the lung cancer risk of coming back and all that.
Yes. That's
Just
correct.
all that and
Yeah.
the pattern, though. So these lymphoma and lung are only two with the highest first year, and then quickly drops fast. So I don't know why lymphoma is more than double in the first year. I think we need to kind of study the biology and understand why why this phenomena happens, and this shouldn't be a lung transplant only phenomena. So I bet some people have described post-transplant lymphoma is a diagnosis. Post-transplant lymphoproliferative disease is a well-known phenomenon. But so we need a dedicated paragraph about that phenomenon.
Okay.
And then skin is more, consistent.
So that should be original article. as I asked you.
So I think there's a lot of, has to be original article. There's a
Yes.
lot of data. So
Yeah.
there's a lot of connection with Dr. Eugene O's paper. So
Mhm.
Jian, you should talk to Eugene because she described with organ transplant
Yeah.
how, there's a contamination, but also immune suppression, and like there's a lot of preclinical papers proving that theory. And we talked about how even melanoma melanoma cells. So I tell this to everybody in my lab, a donor, let's say these are deceased, but if you have a living donor, let's say a kidney transplant, donor had melanoma resected, like Mohs surgery, resected, nowhere else in the body. And then you have the DNA profiling of this melanoma. And then this person gave kidney to, another recipient, and the recipient develops melanoma. And they sequence that melanoma. It's the same melanoma from the other person. Okay. Do you know how that happens? It's immune editing. That person, although it's a localized melanoma, the tumor cells may be still circulating in the body, but never able to grow because of really good immune system surveillance. But in the kidney, kidney is hypervascular. In the blood vessel, there's melanoma cells in there circulating. So it gets transplanted together, okay, with kidney. And they have different, the donor recipient has different immune surveillance system. And it's weakened by lifelong immune suppression. So there can't be, a good immune editing happening. So that's why the same melanoma can happen. It's very scary thought, but I think it's reality. That's why, Liam and I have been trying to implement prospective screening of all transplant or blood transfusion. Imagine this can happen with blood transfusion. And we've seen, I think, I haven't had chance to explain this to Dr. Jian and other members, but we have really nice paper showing that in all Korean women, this is National Health Registry. Maybe Dr. Jian's data is in there too. So giving first childbirth, and then they can have episiotomy or placenta problems and require transfusion during the first child delivery. So we looked at 1 unit, 2 unit, 3 unit of blood of these patients, not even patients, these are mothers. They don't have prior cancer or blood disorder, and they have very tight dose response. The more blood you receive, the more cancer they get in next couple years. And these are lung cancer, brain cancer, pancreatic cancer, and it's scary to see the data. And this, to me, confirms that through blood, a lot of young male smokers, they can donate blood, red blood cell, and those blood can be transfused into the first-time mother, and that mother can develop same lung cancer. That was under immune surveillance for that smoker who donated blood, but in pregnant women, their immune system is suppressed, okay? The fetus is foreign. So the mother need to have immune tolerance to fetus. So their immune system is not same. What I'm saying, in the pregnant women, their immune system is suppressed. So what I'm saying is postpartum is not that much different. I may argue, but anyway, the data is alarming. and we want to prospectively screen patients. You know, we screen blood, donated blood for HIV and Hep B, Hep C, but we don't screen it for cancer. Now we have technology to screen it, but we don't do it. So our lab's been trying to make that happen, but it's not easy. And Liam knows how statistically it's not easy, and funding wise, it's not easy. But this kind of goes back into that same theme that our lab's been working on. So work with Eugene, read all her papers that she has written so far. So that will help you write this paper as well, Jian. Okay, so a lot of thoughts, right? Okay, next. I agree, you can write these pattern, and this could be a nice abstract.
Yes, okay. Andrew, can you unshare your screen? These are the table, and when you look at the table 1,
Mhm.
male would commonly get post-transplant malignancy, but I didn't perform the analyze, but when you look at the trend, it is double in male, and it could be also interesting. And the 5-year overall survivor was higher in cohort who had a post-transplant malignancy, just like a graph. And this was the cancer type that Andrew showed you. The three is I'm still thinking about what to be. And this is a supplementary table, the indications for double lung transplantation. It was not that different, but I will compare between 2 groups. But because the factors are so many, so I want to put them in supplementary.
Yeah, supplementary.
And maybe I can choose the top 10 indications. This is too many. And also there is cause of death. Also the variables are so many, but I can sorting out the graft failure. And I can make another table or graphs with this table, this data.
Yeah, and then, you know, what is the chance of having graft failure and cancer? You know, you can look if there, you know, there probably isn't much happening together, but graft failure and post malignancy, you can put like a Venn diagram.
Yeah.
And then yeah, and then you can do patient who eventually develop cancer versus not, and time to graft failure, you know, see how the graft
Oh, yeah, okay.
changes. Yeah. And you can do it the other way, you know, graft failure, yes or no, and then show how the cumulative instance of cancer changes the graph.
All right.
Yeah.
It could be interesting.
But whatever you have up to today, you can write it for ASCO.
Yeah, I have a lot of research already, so I
Yeah.
can make a short abstract.
Yeah.
And the ctDNA project.
And again, if it doesn't get accepted to ASCO, then still write the manuscript, and whatever you have until I think April, we have World Lung in Singapore. Yeah, and maybe you should come to Singapore to present. I know it's not breast cancer, but
Yeah.
you need a break.
Yeah. Yeah.
Okay.
Jisan will be close than me.
Yeah,
Singapore.
Singapore is closer than here, so.
Yeah. Because we will be in Korea at that time.
Yeah, you'll be in Korea at that time. It's in September, so.
And the next project is ctDNA. Yesterday, Lou sent the email with their raw data. I
Mhm.
didn't look that yet, but only blood NGS results are completed, but the tissue NGS, there are still 5 or 6 cases they need to work on. And Lou said they can give us back by March, mid-March.
So tissue NGS is there to confirm.
Mhm,
So
mhm.
once they double check the blood NGS,
Yes.
we get it and we
Mhm.
stop the analysis. Yeah.
Yes. So I think we can see that first. And
Are you gonna screen share?
oh yeah, here. They gave us the data. And yeah, I need to study. Yeah, I will summarize and I will tell you.
So this is what they sent you, sample volume. So where is the level, their final score? Copy C and B, copy number. Tumor fraction. Yeah. So yeah, this is the final. So at least you have.
Yeah.
Yeah, tumor fraction there. Oh, there's a lot of zero. So do we know what's baseline and patient 1, 2, 2, 3, 5? There's no subject ID. So how was it?
Yeah, here.
Oh,
I
diagnosis
think yes.
after operation.
Yeah.
Wow, there's a lot of zeros after operation, after during chemo clearance. Okay, but also during they have some. Very interesting. So even at diagnosis, plasma, it's not able to detect it well.
Yeah.
So let's see. At diagnosis, it's just
Only 1, 2, 3, 4, 5, 6 patients.
wow. So the other ones are not evaluable.
Yeah, this is during neoadjuvant. Only 2.
Only 2.
And okay, after neoadjuvant chemotherapy, only 1.
Only 1.
After surgery, should be 0. Yes. Yeah.
You did good job.
Yeah.
Dr. Jill.
Yes, I think so.
Yeah.
All
After
of my
surgery,
patients survived.
yeah, you cleared it.
Yeah. Thank you. So at least I'm happy.
Yeah. Well, I guess the lesson is there definitely was tumor at diagnosis, but this test is only able to detect 6 patients out of how many?
34.
Okay. And I guess this, if it's 0, 0, 0, but I'm assuming zeros will always be zero all throughout. But like, we can do in-depth, draw a graph for all like 6 patients, what actually happened
Mhm.
and describe. And we might just need, if we're gonna do TIL, we might just do H&E for the 6 patients. And kind of, it will be more like, we're not looking for statistical power, but really describing what's happening. And then maybe enough.
First of all, I will prepare the clinical data versus this result. Because I have already data with the recurrence. No one died, but they had recurrence.
Once you, have the tissue and I don't know, because we decide to send to look at the TIL anyway, right? What's the verdict? Are we able to look at TIL or not?
Yeah, I need to ask to Sujang. Yeah,
Oh,
yeah, I sent it this, but she was so busy to making abstract because due to ASCO, she
Oh.
has only 11 hours.
So
So
she gives you green light, we will still send it to, but we can decide whether we need it for everybody. But I think we need it for everybody because ctDNA TIL may be showing something very different.
different pattern.
Yeah, so I think we still need to send it to everybody. When you get a green light, send it and
Mhm.
combine all that. We should combine all that into one abstract and decide where to submit.
Okay.
But yeah, it's coming. I'm glad to see our first
Mhm.
ctDNA
Yeah,
to life.
and do you want to take a look about the dynamics of ctDNA?
Yeah.
Okay, briefly, I will make it. I want to skip this basic. And the definition of ctDNA dynamics means the real dynamic changes during the treatment or the after treatment and follow periods. So it could be categorized as detectable versus non-detectable, or positive or negative, or we can calculate the delta value between two specific period. So this specific changes may reflect the clinical outcomes. So there will be so many scenarios in ctDNA dynamics, but in this article, they summarized very briefly about these graphs. So when you look at one by one here, 2 graphs, they showed both early spike, but the difference was clearance versus no clearance. When you look at the left one, the ctDNA was detectable initially, and immediately after initiation of treatment, the ctDNA showed early spike. But this is not the recurrence because of the treatment. They
Right.
showed the treatment response. Yes.
Right.
And during the treatment, it decreases, and after
Clear.
treatment, it became non-detectable. So, but with the right one, there is only spike. So that means that there was a treatment response, but during the treatment, it didn't become non-detectable, and it showed still detectable, but and it showed a plateau. So even ctDNA still shows some values. So this disease will show poor outcome.
Yeah.
And yeah, next 3 graphs, they show no spike, but difference is clearance versus increasing versus no clearance. Initially, ctDNA were all detectable, but when you look at the first graph, it decreases during the treatment, and after treatment, it became undetectable. So this disease will show good prognosis. But the center one, during the treatment, the ctDNA increase. and even after treatment, it increased more. So this would be worst scenario in clinical world.
Yep.
And the right one, there was no spike and no clearance. It just showed plateau.
Yep.
It could be, yeah, worse outcome. And the next one too, our difference is the time of spike. Left one showed only spike because of the treatment response, but the right one showed a late spike because of the resistant to treatment. So left one definitely because there was a treatment response, but after treatment, it's still ctDNA was detectable. So it could be poor prognosis. Right side, during the treatment, it showed the latest spike, which could be the resistant to treatment. So it could be worse outcome than the left one.
Okay.
And this article shows various analysis method, I mean the graphs. So that's why I reviewed. This study was done with the colorectal cancer with the liver metastasis with 54 patients with 380 serial plasma samples. There was a two cohort, cohort one, okay, look at this. Cohort one was the patient who received the surgery first and then get the adjuvant treatment, adjuvant chemotherapy.
Mhm.
And cohort two was the neoadjuvant chemotherapy group.
Uh-huh.
And they used a tumor-informed personalized approach for ctDNA analysis, where at least one somatic mutation was firstly identified in targeted sequencing, then the presence of the same mutation was then assessed in plasma samples. So they compared both the tissue and plasma. During the neoadjuvant treatment, there was 4 cycles. At the first cycle, ctDNA positive group was 91%, but after 2 cycles, it becomes 75, and after 4 cycles, it became 29%. It decreased.
Hmm.
And they also show the values. This could be the one another graph during the treatment. They showed how could it be different with the different analyzing method.
Mm-hmm.
So finally, every patient showed an detectable ctDNA research.
Mm-hmm.
And they compared the 5 years recurrence-free survivor, and T0 negative means that initially the ctDNA was not detectable
Uh-huh.
versus ctDNA clearance group. And the second one is versus no ctDNA clearance, which means it's still detectable. Interestingly, when the patient showed the clearance, the survivor graph were different, significantly different between two groups.
Oh, so no clearance at C4 is not significant?
Mhm.
I guess it's just the
Yes,
sample size, right? It looks different to me.
yes, I think so.
Yeah, I think it's just a sample size issue, but they
Yes.
look similar.
And this could be another way to expression. This is Sankey plot. We can categorize as a positive group and the negative group with the dynamics. We can each line means each patient's research,
Uh,
and so maybe we can make these graphs as well. But yeah, also are negative.
oh, I wonder why they colored that this way. Why did they color red versus purple?
Uh,
Oh. I guess just, they don't, it doesn't mean any.
Oh,
Right.
no, cohort 1 and cohort 2, I think.
Oh. Oh, okay, so the cohort 1 is adjuvant, purple,
Uh-huh. Mm-hmm.
cohort 2 is neoadjuvant.
Neoadjuvant.
Oh, okay. So even that neoadjuvant, there's only 31% that were, well, I guess, no.
No,
Um,
many patients show the ctDNA positive.
mhm. And then it persisted end of adjuvant, neoadjuvant.
Yeah.
And it recurred. Why? So I don't understand. In the neoadjuvant, oh, the positive patients have recurred, but who cleared did not recur. So Xian, we don't have recurrence, right? Everyone is cured. Do we have recurrence?
Oh, I think 1 or 2 patients recurred.
Oh, okay.
Yeah.
So I think we could do similar things like a neoadjuvant, ctDNA, end of neoadjuvant. And then we can put not recurrence, but we can put PCR yes or no, PCR. And then
Yeah,
we can have
okay.
the next column to be recurrence, even if it's 1 or 2.
Mhm.
So that way we can draw something similar like this. Yeah.
And these are just the recurrence-free survivor and overall survivor.
Hmm.
ACE is recurrence-free survivor, and BDF are the overall survivors at each time point. And this is swimmer plot. They expressed red spot as ctDNA positive and green spot as ctDNA negative, and it shows very clearly at each time point
Mhm.
whether the ctDNA was positive or not negative.
Yeah, I think this graph is good. We need to, I think for this, we have recurrence-free survival. Only 1 to recur, then, it doesn't make sense to draw this. I think we can do, so, some people have 100% PCR, but some people it's 80%. Do you have that percentage, right?
Yes, I think
Major
so.
pathology, like a response rate. So we can put that on the what, that instead of the RFS, we can put pathologic response rate on the right side. Yeah, I think that would be good. Baseline,
So
oh, wait.
that's all I have to do.
Go back.
Okay.
So baseline, this is we have only 6, we can't, it's hard to do that. Baseline post-transplant is what's most important, but again, we only have 2 patients post neoadjuvant, so I don't think we can draw that. So just per 1 patient, every patient we need a graph of how it changes. So that's VAF on the right y-axis. Okay, very good. Very good.
That is all I have today.
Yeah, so maybe next meeting you can let us know how those graphs look on our patients. The question for Predison is those that were detected to have mutation, What are those mutations? What gene, what variant? We can ask that, we can ask that. Um, and were they present in the tissue, right? So we need
Mm.
tissue for that.
Mhm.
Yeah, I think it's straightforward. I think it's straightforward. What's where should we present in your mind, Dr. Chiang?
Well, this project, this abstract,
Yeah.
well, uh, my medical oncologist will decide it. I don't want to.
Let medical oncologist
Sorry.
decide.
Yeah. Yes. Yes.
Okay.
Yeah.
ASCO would have been really good if we had this result like a month ago. ASCO would have been good, but
Yeah. Yeah. I think so.
because you can.
Or the next year, maybe. EACR, ASCO.
Yeah, next year AACR with everything. Yeah.
Yeah, and cancer conference. It is
Mm.
big.
Do you go to breast cancer conference?
Yeah, mostly.
Which one do you go?
Mostly ASCO and sometimes San Antonio,
Mm.
and sometimes St. Gallen in Europe.
Where is that? Say.
St. Gallen. But the conference is held in Berlin.
Oh, Berlin. It's European. Mm,
Yes.
okay. Okay, we'll think about it once we will have all the data analysis and everyone agrees. So thank you. Haesung.
So you are
Oh.
muted.
You are muted.
Oh, I'm sorry.
Oh, I'm sorry. Yeah, sorry. So this week, just did some little stuff like with the mRNA oncocyte project. It seems like we just got during this meeting, we just got a reply from oncocyte, and it seems like it's gonna be tough to work together. And so this week I looked into TIDE, which stats just presented. I'll also look into other like algorithms that you talked about, that's available on Tempest. And then I also helped Dr. Jian with the double lung transplant. Besides that, for the DART, the second trial, it's just minor things. The email said like I should add Dr. Kursrog and Dr. Patel. So I just, created these like separate tables and copy and pasted.
Okay.
She also mentions Megan Otis, like talking about her in the paper, but I'm not sure where exactly to mention her name. I'm not sure if I should skip it or use it in somewhere.
Add Megan.
Yeah.
There's no spot.
No, I
No
mean
spot.
at the very end, there's this, but I don't think this is what she meant. This is reviewers. I don't think so.
Then just under that investigator, just put primary statistician Megan and
Oh, like
yeah, like below. Yeah.
I guess I could do like E1, E2, and then okay, yeah, I'll figure something out. And
Yeah.
then another big question was Dr. Kursbach wanted to know if you wanted if you wanted to keep the same cohort, all these 52 cohorts.
I'll say, well, it's yes for now, but maybe Haesung, we can sit down and go over all the pathology together. But for now we'll leave it, but yeah.
Okay. Okay.
So
When
maybe
should
Haesung,
we-
if you can look at like there's a EU rarecancer.net or something. There's a lot of rare cancer related committees or associations. Maybe they have a like nice rare cancer list. If you can look online or literature to find a good rare tumor categorization, maybe we'll apply something different this time. So maybe
Why? What was the issue before?
we the issue is we have low accrual rate for a lot of them. There's ultra rare, some of them. So maybe we need a better category.
And the one before was based on WHO, I heard.
Not necessarily. It was more that EU thing. We looked at it, but there may be a better one.
I guess like another question I had before was the small cell ovarian cancer hypercalcemic type that was actually part of non-epithelial ovarian cancer. And so I was just wondering like why we would separate that out and put it as like a separate cohort. Like is that some of the things that you were also like wondering, like whether we should feel each one as a separate cohort or as like a one big non-epithelial cohort?
Yeah, what in the how big should the category be?
Yeah,
Or
I
do
see.
we have a separate one? So it's a hard decision. It's small cell we added because of the New York Times article. And non-epithelial has so many different biologies inside.
Yeah, so many.
So it's hard, but you have neuroendocrine of breast, neuroendocrine salivary gland, you have neuroendocrine of prostate. It's everywhere, but we lump them together as a one neuroendocrine cohort. So and then you can have anatomic vulvar cancer, penile cancer, you can have all these rare anatomic things. But within that anatomic, you can have squamous, you can have different rare types. So it's hard. It's a very difficult decision.
You're trying to balance between like getting enough sample size versus like controlling the heterogeneity of the cohort
Right.
population.
We want at least biological homogeneity.
Yeah.
Yeah.
Like by biological, you mean by like at least similar pathophysiology, like similar biomarkers, kind of things like that.
Yeah.
Okay, yeah, I'll look into that today, and then maybe we can discuss today or tomorrow.
Yeah.
Okay. Wait, where's my cursor? And then, oh yeah, the Tempest. So yeah, I actually got access to all the Tempest data. This is regarding the TAP THR project. I was very confused because after applying all these filters, which includes the squamous cell lung cancer, who have been treated with immunotherapy, and then I asked for all these modalities like clinical DNA, RNA imaging, and then I applied, non-small cell cancers, and then so after applying that, so basically I was given access to all of their data, and then I applied these filters, and it still leaves me with 1153 patients. So I was confused because according to their like IRB restrictions, I was only supposed to get 499 patients.
Right.
And so should I talk to Tempest about this? Or should I just take out, you know, the ones I won't want? Yeah. I don't know.
So I know for formality, it's 500, but I don't know, just ask like, this is the patient we want to look at first. Can we get all the data and maybe and then once we realize what's more important, we may only publish, you know, 499, but we also at least we want to be able to kind of explore the data set. Because, you know, because randomly selecting 500 out of 1100, you know, it's your luck. Like, what 500 are
Yeah, and there's like selection bias too, I think. So I
right?
don't think we
So
should do
maybe
that.
explain that. We don't want any bias to be introduced by just randomly getting 500 out of 1100. So is it okay if we analyze the data first and figure out what to do next, or what, you know, kind of. Oh, here you have gemcitabine only 2. So some
Yeah, so that's why I thought it was weird. I applied all the filters, but I'm still getting like chemo only patients.
Yeah.
Also, when you look at the histology, I chose squamous, but I got adenocarcinoma data as well. So many questions here. I guess I should just
So,
talk to Tempest
the
about
answer is
this.
their data is not clean. No one really cleaned their data. So the way they get their data
Mhm.
is through AI text mining.
Mm.
So they let AI figure out, but it's not perfect.
Actually, here, one big question is we only want patients who are treated with immunotherapy as their first line treatment. But
Right.
here, yeah, that's a big thing. But there's no way of me knowing this. I don't get to add, you know, like the lines of therapy. I'm just not given that option from what I know. Like regimen is just, I think it's just the name.
I see. There's no. I see.
Yeah, that's a big thing. Also, we wanted to analyze blood TMB data, but I think this is only tissue because that's the main thing, right? And so I think this is just tissue, but I mean, they didn't specify, I'm just guessing. And so we have
TMB.
TMB 258 data.
Oh, out of 1100 TMBs
Yeah,
available,
1153. Yeah, TMB
only
only
available in 258.
Oh, that's true. So we can only run the analysis on these patients, but then that also includes like chemo only, and you know, like the data isn't really clean right now. What should we do?
So first question is, I thought they would have more lung squamous with TMB available. So our question is, hey, is this really true? You guys from the whole US ran you know, NGS on 253, like meeting this criteria. It seems like a small number to me. So is this the right, do you think, you know, just ask that question.
I mean, would you like me to try including adenocarcinoma so that it's like a bigger pool of like non-small cell lung cancer? And then see, you know, how many TMB. But then, you know, there are other questions too, like what the lines of therapy and stuff.
Right. So ask if there's any way to distinguish. They said no. Then we're kind of stuck. Then we have to go with what they have. So if you add adeno and get close to 500, then we need TMB for our analysis. So yes, we need to work with that. Non-small cell. And because we are interested in outcome using immunotherapy, I feel like there's a lot of chemo and targeted therapy listed. because you did select for immunotherapy only, right? It was a filtered.
Yeah, it was filtered.
That's the next question. Like, I filtered for immunotherapy, and the data I
Right.
look at is only. Why is there so many chemos and? So the question is that the therapy that they collected, but because, that patient may receive 3 lines of therapy and maybe the chemo portion was recorded, but immunotherapy was given at some point, but it wasn't
Oh, I
recorded.
see.
So does that mean the patient got immunotherapy at some point, but the recorded therapy was chemo? So there's a lot of questions.
I see. So like overlaps, like does it list all the lines of therapy, right? Okay.
Right.
And would you still rather prefer to have patients who are treated with immunotherapy as their first line? Because I know for Oncocyte, you said you allowed first, second, or third, just in case we miss the EGFR positive patients who were first treated with targeted therapy, but didn't respond, and so we tried immunotherapy later.
Yeah, I think we should just cast the wide net here first because data's not clean. So maybe for second, third, anytime immunotherapy, we'll collect and kind of see what the data is. We don't want to exclude them first, but I'd like to know the first line result. But again, it may still be limited, so.
Okay, yeah, I'll ask them a bunch of questions. And then besides that, I just did some journal club, but it seems like 12:30 already. So, I mean, I can do this next time too.
Yeah, let's do it next time. Today we have the abstract, so
Yeah.
I'm there.
Yeah, thank you so much.
No problem. Liam, I still want to go over any items that you may have.
Yeah, nothing much from me. I was able to submit all of the abstracts for ASCO that I was in charge of yesterday. So the 2 DART abstracts are submitted. That's the desmoid and the vulvar cohorts. The lung cancer, the BAC cohort, we're going to aim for world lung because of the limit of how many you can submit. And then the DREAM study we submitted as a trial in progress. Yeah, so those 3 are submitted. And then I'm just following up with a couple of our pacemakers videos. We're in the process of finalizing editing for Andrew, and then I also received them from Shauna, but she wasn't able to upload them, so I'm gonna have to work with I'll probably work with Eileen and Grace King to get those uploaded in the next week or so.
Oh, okay, but she edited it, it's final?
Yes, we just have to make some final touches. So apparently she couldn't do the captions and like subtitles with either with her computer, or they wouldn't, like the program wouldn't allow her to upload that version. So we just have to finish adding the captions and subtitles.
For Dr. Kim Bomseok, can you see if you can expedite that one, because I'll be actually visiting. So if we can get his work out in next 1 week, that would be great.
His pacemakers video, right?
Yeah, prioritize that one first.
Okay.
Yeah, so there's a 3 interview from Korea, right? 3.
Yes.
And then Andrew did Dr. Mitchell. And anything else pending?
And the Grace King got in touch with Emily Tanez. That interview is going to be this week.
Okay. Okay. And you know, we have Sarah Fenton, I have Pat Mall. So the
Eileen's been
providers.
trying to reach out to Pat Ball, but she hasn't.
Oh,
been-
I was gonna. Oh, we scheduled an interview with her.
Oh,
With
perfect.
Pat?
Yeah,
Okay,
so it's gonna be on February 21st. So next week.
okay.
Sounds good. And I think Casey can do Sarah Fenton potentially.
Yeah. Casey, you should do one before you leave. Yeah. And who's doing Sarah Kang again? Because we decided to do it again.
Oh, we haven't. We're scheduling that with Sarah. Whenever has time, I'll find someone.
Hmm. Okay. Okay, how is the pacemakers coming along, the paper?
The manuscript. So I just have to finish up some parts of the results section.
Hmm.
I would say it's about 75%. Like the draft is about 75% ready.
Okay, and that you wrote like the both themes, like we had two posters.
Yes, we combined it. Yeah.
Combined it, yeah. Can you send me those two posters again, just so that I have it in file, right, somewhere, but yeah. I wanna
Sure.
review them again. So we need to work on that. Okay, and the Dart. So we have ACR is not out yet.
Yeah, the email should be coming to you. So if you haven't heard from them yet,
Do you know
but
by when?
they said the 16th, so this Thursday.
On this Thursday, okay. Okay, so 16. So again, Liam and Haesung, I want you to work on these start cohorts as they come. So so these are, I consider, so thyroid will be a good paper because we're gonna, oh yeah, so that's the follow-up. What happened to, because we were planning to do follow-up with like the NGS result with Kara Haymaker.
Yeah,
So can you follow up on that to see
sure.
when that's going to happen?
Yeah, I know we have a meeting. We had a meeting coming up. I think it got rescheduled.
Okay. Yeah, just follow up with that. And Haesung, you can be involved in. So Haesung and Liam, you guys are both in charge of writing the results for the Dart. And I think the priority is thyroid because it's going to have translational medicine piece, but the salivary gland, small bowel, write them up together, okay? And then once you're done, then continue writing up the next cohorts. Those are gonna be brief reports, like, you know, it just adds a little bit more to the abstract. And the Grafton tables are all generated by Megan. So yeah, so I want you guys to work on converting them to actual paper as in the order I described. So that's just another thing. Anything else? So
No.
again, follow up on the biomarker piece, the budget, and we're still kind of in limbo. So yeah, follow up on all the papers, companies where things are, and yeah, we really need to yeah, work on that. Okay. I think we're good for now. I know we haven't had to go to the students now, but we just were we have deadline today, so let's work on those and see what we can do. Yeah, and then I guess because today is deadline, I think we can skip today's so today, which book are we doing, Liam?
So it's supposed to be 다시 만나는
QA. Okay, we can do that
tomorrow.
Wednesday.
Yeah.
Because today's dead, and then inspired we can do it next week. Because I know you guys have deadline to work on that, so.
Sounds good.
Okay, so any questions before I wrap up? Anything? Okay, we'll wrap up here, and we'll see you guys later.
Thank you.
Yeah.
Thank you.
You.